is, now in obesity, the mitochondrial dysfunction is a key factor. Hello? Can I go ahead, madam? Mitochondrial dysfunction is a very, very important function. So in order to look at the mitochondrial impact factor, so we know that we have now targeted, because in 2007, we have come to know that in all of the target organs like visceral and subcutaneous, you have what is called as brown-like adipose tissues. Earlier, the brown adipose tissue was present only in the newborns. But now, after 2007, it is now come to know that these adipocytes play a very critical role. So this is the white adipose tissue, the subcutaneous or the visceral, and this is called as the brown adipose-like tissue, which is able to get itself activated under energy homeostasis. So now we have actually targeted this pathway now, and if you look at this slide, we got very good encouraging results. So the placental mesenchymal treatment for 8 weeks, and after the 8th week, when we take out the target organs, the visceral tissue, the adipose tissue, both subcutaneous and visceral, we found the important markers like PRDM16, the UCP1. All these are important markers, which is much, much lesser in that without treatment, suggesting that the stem cell treatment, in addition to the insulin signaling mechanism, it is also able to upregulate the energy homeostasis in the body through browning of the white adipose tissue. So please note that browning of the white adipose tissue, this is a very important area of adipose tissue biology, which we are now working on, and very good leads we are getting. And if you look at this slide, you will understand that the mitochondria content has decreased in these obese animals. So when there is a decrease in the mitochondrial content, the respiratory rate is decreased, they are more lethargic and more prone for infection, because the oxygen circulation is very, very vital for any target organ. But interestingly, if you look at these two, that is the dark where my arrow is actually pointing out, both in the subcutaneous and in the visceral, the intensity is very, very dark, and we are able to show the increase in the mitotracker. It's a mitotracker, which is a mitochondrial signaling mechanism. Right? So we are able to demonstrate that with mesenchymal therapy, the obesity management could be one of the mechanism could be by targeting the energy homeostasis in addition to the insulin signaling mechanism. So this is what we are now putting the, very open, because the browning of the adipose tissue is brought by several nutraceuticals. Can we also include the, the mesenchymal stem cells in the list? It's definitely, we are working more into that before we can, comply saying that it is one of the important molecule. And can we also increase this as a, for expenditure of energy? That is going to be our target of research work. And just yesterday, my student, Dr. Samuel, he gave me a very good paper saying that already in nature science, the nature nature communication, it has already come that these your stem cells are able to increase your beiging property, and that is going to be the target. Or for the next few years, you are going to have the B cells injected into the body, and that can help you in the reduction of the visceral fat. That is going to be one of the target, which is just published in 2020, and that is in the Day for Yesterday's issue. And this is the chapter, Human Beige Adipocytes for Drug Discovery and Cell Therapy in Metabolic Diseases. So we are very, very excited, because we are moving the right line of direction, where we show that the mesenchymal cells, in addition to their inflammatory reduction, they are also able to have an impact on the energy status. So what are the important leads that we have got till now? The important leads we have got in the management of obesity, especially in the preclinical approaches, we are able to reduce the glucose toxicity, the lipotoxicity, and also the important trophic factors are able to have an impact or a bearing on as a drug stores within the target organs. So this is what I want to know. But madam, I just give me some few minutes time where we want to show the stem cell therapy in the osteoarthritic management, which my student inside will give just a 5 to 7 minutes presentation.
Please, okay, go ahead.
Yeah.
Very good morning to everyone. I want to thank Madam Dr. Vijayalakshmi Venkatesan for giving me this opportunity to briefly explain about the kind of work that we are doing in our laboratory with applications of stem cells, mesenchymal stem cells in the field of osteoarthritis. As we all know, osteoarthritis, it is a condition of the joint, in predominantly where the cartilage tissue is lost, is deteriorated, and eventually what happens is with the loss of cartilage tissue, the bones begin to rub against each other, and this causes stiffness and pain, and there is a limitation in the movement, in the joint movement, and that is why it leads to morbidity and eventually mortality. So osteoarthritis by far it is the most common type of joint disorder. So to give a little statistics, in India, osteoarthritis, the prevalence rate is somewhere between 22 to 39%, and it is more common in women than men. And the most common types of osteoarthritis are the knee, the hand, and the hip osteoarthritis. So close to 45% of women over the age of 65 have the symptoms, while 70% of women over 65 years have, they show radiological evidence of osteoarthritis. So the burden of the disease burden of osteoarthritis is huge, especially in a hugely populous country like India. So what is to briefly give you the pathophysiology or how the disease progresses, exactly we still do not know what is the initiating factor. But what happens is, because of obesity, the overweight, the load-bearing joints, the knees, there's initial trigger where the cartilage breakdown happens, and this sets down a battery of, reactions, which lead to, one after the other, causing the joint failure. Chiefly, the pro-inflammatory cytokine called the interleukin-1 beta, it is capable of inducing the chondrocytes, and then, so because these chondrocytes, when they get stimulated, they synthesize this matrix metalloproteinases, which are the key enzymes which are involved in degradation of the articular cartilage. Eventually, what happens is this interleukin-1 beta, it also inhibits the synthesis of collagen 2 in chondrocytes, and eventually, when collagen 2 and these proteoglycans are not synthesized, they, obviously the extracellular matrix is not retained, and eventually it leads to the death, the chondrocytes death, and finally leading to the degeneration of the articular cartilage, finally resulting in osteoarthritis. So this is just a brief illustration as to how the cytokines can act upon the chondrocytes, which can give rise to this matrix metalloproteinases, and increase in enzymes, and the reduction in agrican and type 2 collagen, which are this extracellular matrix component, and finally resulting in the failure of the whole joint. Again, this is a complex schematic schematic illustration to show how mechanical stress or the cytokines, chiefly the IL-1 beta and TNF alpha, through transcription factors like NF kappa B, can act upon not only the chondrocytes, it can also act upon the synovial macrophage, the synovial fibroblasts, the subchondral bone cells. It can, you name any type of cell, in all the cells of the joint. These cytokines, through transcription factors and other components, they have a complex network of reactions by which the chondrocyte apoptosis takes place, eventually leading to chondrocyte death and the failure of the knee joint as a whole. So, as Madam was already been explaining, we have also been working on this WNA GROB, the obese mutant rat model, to see if we can have any spontaneous onset of osteoarthritis. As we see here, this is a control Vista rat at different age points, 3, 6, and 9 months. But here, Vista rats at 6 months, at the GROB rat at 6 months, we see that there is cartilage erosion. And at 9 months, the whole joint is completely collapsed when you compare with the Vista rat. So even with the grading for cartilage and the subchondral bone, we see that with increasing age, these rats, these GROB rats, they show progression of onset and progression of osteoarthritis, the cartilage tissue. So this is a scanning electron microscopy analysis of the tibial cartilage tissue in the Vista rats and the control rats and our animal model of study. If you see here at different times, at different age groups, 3, 6, and 9 months, the surface is smooth. The cartilage surface is smooth. But in the GROB rats, with increase in age, as you see it, especially at 6 months and 9 months, there is, starting from 6 months, we begin to see that there are degenerative changes happening in the surface. At different magnifications, we see that, especially at 9 months, we see the whole cartilage is eroded, which is very closer to the human scenario. So again, this is micro CT analysis of this subchondral bone. So not only the cartilage tissue, even the subchondral bone also has its impact. Because when we see here shown by these red arrows, we see that in the GROB rats there is increase in the subchondral sclerosis. That means the subchondral bone begins to thicken. So in fact, indirectly, it hampers the health of the cartilage, both biologically as well as mechanically. So, it's a complex, both at the cartilage level and the subchondral bone level, we are able to see, changes, pathological changes which closely mimic to human osteoarthritis. So, to confirm at the cellular level, as I already told you, collagen 2 and MMP13 are important markers to see the progression of osteoarthritis. As we see here, when we compare at the 6 months age, we chose this age group to see what is the cellular expression of this collagen 2 and MMP13. At 6 months old, the Vista rats, they showed up, very prominent expression of collagen 2, but this our experimental model at 6 months, they know that the intensity of the collagen 2 is less. Similarly, this MMP13, what happens is at 6 months old, this Vista rats, the MMP13 it is a proteolytic enzyme that the expression is very less, which means that the knee joints are, the cartilage tissue is healthy. But we see that at 6 months at GROB rats, the cartilage expression of the chondrocytes expression of MMP13 is very high, which means that there is indication of the onset and the progression of osteoarthritis. So to show the various stages, it's a cartoon to show, the mild stage. What happens is the cartilage breakdown. This is smooth, but we see that here and there are some mild cracks that are beginning to happening. And at the moderate stage, what happens is the synovial, the joint, this is a femur and the tibia. We see that the joint begins to undergo that degenerative changes where there is cartilage erosion, and these small osteophytes, these are the bony spurs which begin to form, and patients experience pain. And in the most severe case, we see that the whole joint, the cartilage is completely eroded, and there's a lot of osteophytes, when this is a stage when, the bone begins to rub against each other, because essentially the cartilage is like a cushion, it helps in weight bearing, and it also helps in the flexibility and the lubrication and the movement. And we see at the advanced stage, the white cartilage layer is completely eroded, and the bone begins to rub against each other, and, patients suffer from immense pain. So these are the current treatment strategies currently available. Depending upon the grade of the disease, the mild, moderate, or severe. At the most severe case, there is no other option other than surgery, because the people have to go for complete knee replacement. But at the mild level, at the moderate level, people go for this physical therapy, exercise, or nutraceuticals, or this painkillers. So there is still, till date, there is no single cure for osteoarthritis. So that is when, because as already you would have listened, Madam has explained about the promises of mesenchymal stem cells, how, because of its plasticity, when you give the appropriate environment on the factors, you can program these stem cells to differentiate into your cell of choice. For example, like the bone cells or the osteocytes, endothelial cells, the chondrocytes, adipocytes, neuron, etc. So our target was chondrocyte, where we wanted to program to see if we can use this plastic nature of this mesenchymal stem cells, to induce cartilage regeneration. Essentially to differentiate them into chondrocytes. So this is a schematic. What happens is in normal MSC therapy, we can inject these MSCs into the knee joint. It's a site-specific therapy where you inject it into the knee joints, and because of their migratory properties, these MSCs can home into their to the site of action. They can get once they home, they can stimulate and begin the tissue repair process. So our experimental model, we thought, okay, why not just give. We wanted to study the efficacy of stem cells. In addition, there's a compound called the stigmasterol. It is a nutraceutical which is found in soybean oil, and it is one of the very important nutraceuticals. We thought this is already shown to have chondroprotective effect. We thought, okay, why not we just go for a combinatorial approach? If we can see if we combine stigmasterol with MSCs and check if the combination of the per se treatments which is having a better outcome, disease outcome. So this is we want to evaluate the efficacy of the treatments in the rat model of osteoarthritis. So this is just a schematic. What happens is we thought we'll go for a induced model, because when you chemically induce the disease in the rat model and to see how well our treatments are working. So this is called the monosodium iodoacetate. It is a salt which, when we inject it into the knee joints, into the interarticular region, it can bring about chondrocyte death. So eventually, we are mimicking the human scenario in that experimental model. So when we induce the monosodium iodoacetate in 1 week of time, the osteoarthritis begins to set in, and followed by which we began to give the intraarticular injection of 2 million cells, and we followed this, and not only the MSCs, we also injected stigmasterol, and the combination of the MSCs and the stigmasterol, and we followed it for 4 weeks, followed by which we sacrificed. So this is an image representing how we give an intra-articular injection between the femur and the tibia and the knee joint of the animal. So at the, to summarize the results, at the end of 4 weeks, we carried out a micro-CT analysis and histopathological analysis. We see here the control rats, it's showing a very smooth architecture of the bone. But in the osteoarthritis, what happens, as I already told you, when we inject this monosodium iodide, it causes chondrocyte, and eventually there's severe erosion of the femoral bone or the cartilage, and further it has also affected the bone beneath it. And when we treated with the MSCs alone, there was some promise. We were able to see some. The MSCs were able to regenerate the cartilage to an extent, but with the stigmasterol alone, it did not have any effect. But when we give, this is a very promising result that we obtained. When we give a combination of stigmasterol and MSCs, we are able to see that there was a very significant improvement in the cartilage regeneration. So this, we mean to say that there are two mechanisms by which this can happen, where we inject these MSCs, these populating cells, they can go and bind to your chondrocyte lesions, and they can bring about the healing process. In addition, the MSCs, when we inject them, as Madam already said, when we inject the MSCs, they also have the factors which they release. So these factors can stimulate the resident progenitors which are within the knee joint, and they can also bring about this healing process. So this is a histopathological confirmation of what we obtained in the micro-CT. So we see here again the osteoarthritis rats. There is a significant erosion of the cartilage, but when we see this MSC and the stigmasterol treated rats, combination therapy, as almost there is a return to the normal architecture. So this brief study of us, we are able to show the benefits of mesenchymal stem cells and the promise of it in treating osteoarthritis. So with this, I just want to conclude my presentation, and I want to thank everyone for this opportunity.
Hello.
Thank you, ma'am.
Okay, thank you.
These are the stem cell approaches, what we are doing. And any questions?
Yes, ma'am. One question from participants.
Yes,
At
ma'am.
which stage of a problem in a adipocyte does the effect of mesenchymal cells work? In initial or in advanced stage?
It works in the initial stage.
Okay. Okay.
It is in the preclinical stage.
Okay. Vanita, Surender, participants, I think you got your answer. Okay, ma'am.
Okay, thank you, Dr. Vijayalakshmi, for your excellent.
Hello. Dr. Khader. Dr. Khader.
Yeah. One more question. Hello.
Yeah. Yes, madam.
Can you hear me?
Yeah, I can hear you.
Yeah.
Geeta, ma'am, you have muted your mic, ma'am.
Can you hear now?
Yeah.
Yes, ma'am.
Okay.
Yeah.
I had already sent to. Anyway, I'll repeat it. This is to Vijay Lakshmi.
Yeah. Please tell me, madam.
Mostly you've been addressing obesity in relation to mesenchymal cells and how it is oriented towards the repair of glucose tolerance versus intolerance. All right.
Yes.
Now, my question is, in today's burning issues of COVID, I'm more interested in understanding how do these cells, especially that you said about cytokines, you said about many of the imbalances of the mesenchymal stem cells, etc. I want to know, are there any studies at all reflecting on infection as a status of stress? I know COVID, probably there's very little data available, but in general, viral stress has a component, one. And two, can you alter the pathway in such a way? Because today we see that there's an extreme cytokine storm, especially of neutrophils, lodging themselves into capillary blood vessels, leading to thrombosis. Is there any thought process? I know there's no evidence, but would you share any thought process of any corrective measures in this direction, Vijay?
Yeah, a very good question, Dr. Geeta, and a very interesting one. The first question, what I would like to address is regarding the viral, during the infectious time, what you are asking me. There is an Israel group, actually, during a paper which has come in, the Lancet, where they have actually, isolated the, the secretome factors from the mesenchymal cells, and they have actually injected into the patients actually, because now, as you rightly told, there is no focused therapy for, for even COVID or any kind of viral infections. So they were able to show that in for 50 to 60% of the patients, the cytokine storm was actually reduced by 40 to 50%, and the patients did not go into further morbidity conditions actually by injecting the cytokine fraction. So that gives an indirect evidence, or you can say a direct evidence, to show that these cytokines are actually able to, quench or target the, the pro-inflammatory cytokines and try to downregulate them. And the mechanism is, still needs to be worked out, because the trophic factors are embedded within the mesenchymal cells. They can go to the place of injury and actually bring the correction. So this is what we are also anticipating, that the low-grade inflammation, what we are seeing in the obesogenic milieu or in a diabetic milieu, what our earlier publications very clearly shows that really it works wonder with respect to a preclinical model. See, my model is a preclinical model. It has not gone to the beyond preclinical, where I'm saying that there is a liver cirrhosis or there is a hypertrophy where I require only an organ transplantation. We have not gone to that level. We are in the intermediate level, a preventive level, where we can actually target the organs where they have not gone into a metabolic, too much of metabolic stress and death. So that is my first answer to your question. The second question is a very interesting question. Is especially among the different classes of mesenchymal cells, the placental or the perinatal cells like umbilical cord or the Wharton jelly or the placenta cells, they have lot of VEGF activity. That means they have the endothelial factor, secreted within them. So suppose I transplant the placental mesenchymal cells, and if there is a block, it is able to negate the block by secreting a lot of endothelial specific factors, the trophic factors, which is not there in the bone marrow, as well as it is not there in the dental pulp derived mesenchymal cells. So what I am advocating is, it is very important that we select the type of mesenchymal cells for a given kind of a metabolic disease or kind of a system what we are addressing.
One more question, Vijay.
Yeah.
One more question.
Yeah.
Suppose I were to give you any tissue from infected patients to identify any biomarkers in COVID-related dysfunction of cytokines or mesenchymal cells, what kind of cells would you dream to have?
I mean,
from live patients, because I'm doing some study.
Yeah.
So, I can share the sample if you can lead into something more interesting.
Yeah, that we can definitely work on. We can work on, because from the biopsy, what exactly, how old is this? The cells are very freshly removed and they need to be put in a very right kind of a medium. It's possible. I'm not saying it is not possible. We can do it.
Can you get into a detailed dialogue with me in that respect, Ji?
Sure, we'll do that. We'll do that.
Okay. Thanks. Bye. Thanks, Dr. Vijay Lakshmi Khader also.
Yeah, thank you.
Vijay Lakshmi's. One Vijay Lakshmi, one Vijaya.
Okay.
Thank you both.
Yes, ma'am.
Okay. Thank you, Dr. Vijay Lakshmi. It's very, very, what we can just say, illustrative lecture, giving all the examples. We know very well what you can say, obesity is the biggest problem nowadays, which is a prerequisite for any type of the illness. So we have to really take about the what you can say, obesity, and you have really explained about it. I'm sure there may be plenty of questions from the audience. You can answer a little later on, and I really congratulate you for your excellent lecture. Thank you very much.
Yeah, madam, one last slide. I would like to thank both the students who are actually with me here. One, my student Surya Prasad, and Dr. Samuel, who actually is working on osteoarthritis. They have been really into this, 24/7 for the work. I should congratulate them, and I should bring them to your knowledge. They have contributed a lot. Yeah,
One
thank
thing I
you.
tell you, any research work, it is a team effort. One
Yeah,
person cannot
24/7.
do it. It's a group of the people. They have to really
yes.
do
Yeah.
it day and night. Then only
Yeah.
you can just show some concrete results. Any
Yeah.
research work, the team of work definitely will be there. I congratulate all your team members also
Thank you,
for
madam.
their excellent work. Okay.
Thank you, madam.
Thank you very
so
much.
much.
Thank you
And
very
thanks
much.
for the organizers for the opportunity also. Thank you.
Dr. Uma, can I move on to the third speaker?
Madam, definitely, madam. Madam, yes.
Okay, I think.
Madam, Sarla Neelam Raj, already I made her a presenter. Now, madam, can you just unmute? I think I'm unmuting you. And enable your video. Can you share your PPT, please?
Can you hear me?
Yeah, very well, very well. Yeah, we can able to hear you and see you, ma'am. Madam, it's shown to you just on the bottom of the screen. You have three icons. Click on the top arrow directly to the top. You can just click on that and choose your PPT to share with us.
Sarla, ma'am. Third
Yeah.
icon is an arrow, upward arrow. You click on that, ma'am.
Right.
Yes, ma'am.
I'm just getting my
Before that, you open your PPT file also.
yeah.
Lalita, otherwise you can open it, ma'am. Ma'am.
Ma'am, PPT is not there, ma'am.
Okay, fine.
Yeah, I'm uploading it. Just a minute.
Sure.
Yeah.
నేను ఆల్రెడీ మేడంకి చూపించాను మేడం ఇంతకుముందు రిమోట్ డెస్క్ లో షోన్ హర్ హౌ టు వర్క్.
Okay, fine.
Yeah, that was nice.
Madam, if any difficulty, maybe you can share your remote desk to me again. I can help you out.
Export my file. I'll believe it is there.
First, open your PPT. After that, you click on that arrow. Then it will ask which file to share. Then choose your PPT. Then it will be done. I know it is. It takes a while for us to use these new tools and everything. So as Vijay madam has already mentioned before, we're trying to learn the new things now.
I'm just wondering how to. Yeah.
Madam, you open the PPT, madam, first. You open the PPT. Did you open the PPT first?
It is on the desktop. Shall I open it first?
First open today.
Please open your PPT first.
Oh, okay, open. Yeah, now?
After that, click on this arrow which I have shown as a third icon.
Right.
No?
Now it's there.
Yeah, great. Thank you very much.
Yeah.
Okay.
Done, madam. Madam, put it in the. Madam, press F5. Press F5.
F?
F5. Punch.
F5. Oh, got
Done. Great, man. Done.
Okay. Should I go?
Yeah. We're all learning first time. Just a second. Now, is this okay? Yeah. Can you hear me? Is the sound okay?
Yes, ma'am.
Okay. Should I introduce the?
Yes, ma'am.
Okay. Dr. Sharala Neelam Raju is currently employed at Directorate of Rice Research, ICAR. She was honored with a gold medal from the University of Delhi DBT Overseas Associateship, received number of awards such as ICAR Senior Research Fellowship, ICAR National Professor Chair, ICAR Punjab Rao Deshmukh Outstanding Woman Scientist Award. She received PhD from Indian Agriculture Research Institute, New Delhi. Dr. Sarla traveled extensively and she is a member of many professional bodies. Recognized scientist in ICAR system with her research expertise in rice improvement. May I request, and she has got 214 publications in national and international journals to her credit. Dr. Sarla Neelam Raju, Please proceed.
Before that, I just want to add one more introduction about Madam. The present Director General is the proud student of Madam.
Oh, good.
He's so proud that Sarala Madam is his teacher.
Yeah, it's really good. It's a good news. Dr. Sharla.
Okay, can I start? Yeah.
Please start.
Thank you, Dr. Vijaya and Dr. Uma, and for giving this opportunity and also for such an elaborate introduction. Thank you. Thank the organizers for this opportunity for me to share our work, which is largely students' work in the last 15-20 years, but I'll be talking about the last 5 years work on chromosome segment substitution lines using wild progenitors for rice improvement. So I'll be talking about rice, but the topic is relevant for any crop. Chromosome segment substitution lines using wild progenitors can be used in any crop. Now, just to give an outline, rice consists of two cultivated species. Oryza sativa, that is Asian rice, has 224 chromosomes, A genome, and there is also the African rice. There are about 22 wild species which belong to 11 genome types. As students of genetics and genomics, we are interested in knowing their genomes. And there are diploid rice wild species and also tetraploid wild species. So we've been interested in Oryza nivara and Oryza ruficogon. These are the two closest progenitors of Oryza sativa, and they have the same chromosome number as Oryza sativa, and they are found naturally in India. Now, just to give an idea, wild species have generally been used as sources of disease resistance and pest resistance, but not much as sources of increasing yield, because the wild species themselves are not high yielding. They are very poor looking plants, but they have genes which can help increase yield. So in the last 20 years or so, it has become important that these wild species are useful not just for disease and pest resistance, but also to increase yield and also sustainability, climate resilience. We'll have changes in the climate happening, flood, drought, salinity, and how can we maintain our yields or increase the yield of rice despite these adverse situations. So people look to wild species. These are the wild species. This is the geographical distribution, and as I said, nivara and Oryza ruficogon are naturally found in the Indian subcontinent. The last 10 years, there have been a lot of papers on chromosome segment substitution lines being developed all over the world using different wild species. But in India, we did not have a set of chromosome segment substitution lines which are useful to map genes for different traits. Now, the wild species have the natural allelic variation. How can we use from the wild species? They are a good source, as I said. And for students of genetics, it is important to know that in the 1920s, sugarcane yield was increased using wild sugar canes, oats in the 1970s, and in tomato in the 1990s. So wild species were used, even though markers were not available, genes were sequencings were not there, but then these were useful in increasing yield. Breeders have been using wild species, but at a low level. Why is our interest in wild species? Because when domestication happened, the wild species to early domesticates to modern varieties, there were genes which were lost because man selected for domestication. So there were genes for shattering and other such undesirable traits were left behind. And along with them, if there were linked genes for high yield, they were also left behind. So these bottlenecks have been there at each step of evolution, and modern varieties also have lost many genes of the early domesticates. So with the advent of markers and sequencing and other efforts, we now can pinpoint the genes from that 400 MB rice genome, which are the genes which are increasing yield, and we can get these alleles from the wild species. And now there are a lot of variation in the wild, not just existing variation in the wild accessions, but there's a lot of de novo variation that is created when wild species are crossed. Now, in 1998, yield quantitative trait locus, yield 2.1 on chromosome 2 was identified, which showed that 20% yield could be increased if we get that allele from a wild accession or rhizosphere. So that spurred our interest in using wild species, and as geneticists, we would like to see how we can help the breeders use this in a very usable way, use the gene pool available in the wild in a usable way. And it's almost known that almost 80% allelic diversity is still untapped. And how do we bring this allelic diversity to the gene pool? How do I go to the next slide? Yeah. So chromosome segment substitution lines have been reported in 22 major crops, cotton, wheat, many other crops, including rice, and almost 75 sets of CSSLs are reported from intra-specific and interspecific process. There's a lot of interest in getting the genes from the wild, and about 46 sets are reported in rice. And these different countries have, but there were none reported from India. So when we started, there are 7 wild species have been used for developing chromosome segment substitution lines, but we started using these 2 progenitor species, and we started with the project on how can we use Nivara rufipogon, the 2 wild accessions, to map QTS, and our interest was on yield-related traits, because there were many groups working on diseases and pests, but for yield it was a challenge, because the wild species themselves are not high yielding, and yet they have genes which can help increase yield. So I'll just, since there are students of bioengineering also here, what are CSSLs? Chromosome segment substitution lines are ideally a set of marker-defined plants, marker-defined lines, which represent the entire genome of the donor parent. So if we take an example, the red is the donor parent, let's say a wild species with 4 example, we'll take 4 chromosomes. We've divided it into 4 parts, and we develop these lines after back process. And how they are useful is I'm trying to show here. Yeah. So if we have 8 chromosome segment substitution lines such that each line has a part of its chromosome segment substituted by the wild chromosome segment. So if we have a whole set, we can have the entire wild genome represented one by one in each line of these chromosome segment substitution lines. So we can study the effect of that segment would have on the traits that we are interested in. So what is done is these, let us say 8 chromosome segment substitution lines are phenotyped for any trait of interest. Example, let us say I look for 1000 grain weight, and I find that compared to the recurrent parent, which is a variety, the lowermost green one, if chromosome segment substitution line 5 and 6 have a higher 1000 grain weight, we can assume that it is in this chromosome 3 and in these segments, because these are the ones which are different from the recurrent variety. So that is the logic of using chromosome segment substitution lines to map traits for a trait of interest. Now, why do we develop CSSLs? Because if you want to map genes to chromosomes and locate where they are, how much is their effect, will they increase yield or decrease yield, we need to develop these chromosome segment substitution. And world over, thousands of CSSLs have been used to map and clone genes for yield-related traits, for 1000 grain weight, for dense erect panicle, for so many other traits. It takes time to develop these back crosses, but once they are developed, it is a good national genetic resource. It is quicker, easier, and more precise. And we can, once we have this set, they can be shared with people. We can genotype them once and phenotype for as many traits as possible. So if someone in the Northeast can phenotype it for tolerance to acid soil, someone can phenotype them for tolerance to floods and such traits. So they become, it becomes easy once such a set is developed. And then near isogenic lines can be easily derived from such a set because they have only small segments from the wild genome which are present. And this will help in map-based cloning and also discovering causal genes for a trait. You can also use two contrasting CSS cells to map QTLs. We can use reciprocal CSS cells to study background effect of the cytoplasm also. And fixed and stable CSS cells, we normally go up to 4 back crosses. They can be shared and they can be tested in diverse. So there's a lot of advantage in having such a set developed. And as some speakers previous also mentioned, even when we do a lot of basic studies, there are always translational spin-offs which are applied, which are of use. And so very often many of these CSS cells, as we have also seen, they can be released as a variety themselves once they are tested, because they are basically most of the genome of the cultivar and only a few segments from the wild, but they are very useful. So this is now of course sequences are available, so people are using SNPs and others, but we use this universal core genetic map of SSRs which was developed. So these are the 12 chromosomes of rice, and these are the markers equally spaced. So it is a very useful resource. We use these markers to look at polymorphic markers which we report. So how we started Swarna and this is Cotton Dora Sannalu, Maruteru 1010. Both these varieties were developed in the Maruteru station of Andhra Pradesh Agricultural University, and these are very popular varieties. So we decided, let us use these varieties, and we chose one accession which was very photosynthetically very high photosynthetic rate, and then we started. We followed the general procedures that have been followed of making diagnosis and for markers at each time. Yeah, so these are the wild species, and these are the F1s. Some of them look exactly like wild, they do look back, but yet they have genes which will help increase yield. And then these are confirmed using molecular markers that they are true F1s. So these are basically our reporting markers from the sativa, and this is from Rufipogon. So we say these are true F1s or not. Now, the program of works is something like this. The wild accession is always used as a male parent because we do not want sterility and other issues from the cytoplasm to affect the recurrent parent, which is a cultivar, and 4 back crosses, and this is a recurrent parent here. So our plan of work was we mapped QTLs in 2 back crosses. QTLs are mapped normally in segregating populations, so BC2F2, and also in BC4F2. There were some lines which were already available with us, and we went up to BC2F8. So we were looking for QTLs which we map in BC2F2. Are they stable in BC2F8 or not? Those are the kind of questions we could also answer, that they are stable QTLs and they can be transferred. Because why do we map QTLs? We are mapping these quantitative trait loci. One is for basic studies to do functional analysis of how they work, and second is to be able to transfer them to varieties. So are they amenable to marker-assisted selection? They have to be stable over generations. So that was the purpose. So this was the scheme. So Cotton Dora Sannalu and Swarna, and a lot of work in getting to these back crosses, and then phenotyping in the field and genotyping in the lab, mapping QTS, and then identifying the chromosome segment subset. I'll just give an example of one or two process. So this is MTU 1010 into Rufipogon. About so many families, 238 F2 families are phenotyped for yield traits. So just see the results. Out of so many, about 34 flowered significantly earlier. So that's a good rate. About 10 families showed a higher 1,000 grain weight. 5 families had higher tiller number, 1 with harvest index and further. And we also measured photosynthetic traits, and net photosynthesis was significantly correlated with many of the yield traits. So these chromosome segment substitution lines, even in BC2F2, had very positive traits, and some side observations that yield was significantly correlated with net photosynthetic rate, since we were doing both physiological studies and field studies on agromorphological traits. There were interesting things that plant yield is also correlated to many other traits, water use efficiency and carboxylation. Again, in BC2F3, so it's not just one generation, we phenotype on 2 generations and then map the QTLs for both the generations. So this is the kind of variation that we observe for leaves, for stem, for seed type. And this is one line which has a very high 1000 grain weight. It has a higher yield than MTU 1010. And it has come to a stage where we entered it in All India Coordinated Rice Improvement Project trials. Our institute is the center for that. And it went into advanced varietal trial for low phosphorus. That means it can withstand low phosphorus and still give the higher yield. Those are traits which come from wild species because they survive in adverse situations. So they have combinations of genes which can help increase yield even under adverse situations. So a basic scheme is, I've just shown a tiller number here for productive tiller number, but this we do for all the traits. So this is the frequency distribution, and then all the lines are genotyped. We have the wild, the allele from the wild and the allele from the variety, and then we see the segregation. So after phenotyping, genotyping, we do a linkage analysis. And this is a typical QTL map for all the 12 chromosomes. Suffice to say that here we found there were 5 clusters of QTLs, and all breeders know that there are many of such traits, 1000 grain weight, yield, biomass, photosynthesis, which are linked, and they are also co-located. They are not just correlated, they are also co-located in the country. So those kind of things one can study, and it's very exciting to know that there was one QTL which was new. It was for 1000 grain weight on chromosome 5, and if it is a narrow QTL, one can fine map it and then go to the gene to look for a causal gene. So these are useful even for gene discovery. Now this is a graphical genotype of BC2F2. So these on the left are the lines. So about 145 CSS cells, and these are the chromosomes 1 to 12. And the red segments are the segments which have come from the wild. The blue is the background. There are some heterozygous segments also there. So if we have a CSS cell of interest, which is very significant for the trait of interest, we can use that and then go for a few more back crosses. So while developing the CSS cells, we can see that in the first BC1, we have almost 20% of the genome from the wild. Then we have BC2 about the same. This is just 39 I'm showing out of 94. So this is how we select the target CSS cells at each backcross generation using markers. And in the end, here in the BC4, backcross 4, we had only 8.3% of the. So we want very little of the wild genome so that the variety is not, the genome of variety is not so much disturbed, and yet we have significant lines available. So these are up to 4 back crosses after we have gone. This is how we test them in field, and you can see there are some very early lines. There are others which are late, and they are fairly uniform by the time they come to BC4F2. The kind of variation that we get for seed type, seed color, and these lines we have also analyzed for iron, high iron and zinc, so this could be useful for biofortification as well. These are the kind of lines and the variation that we get for height, tiller number, seed type. And so almost 99% of the wild genome, as reported by these 160 markers, was represented in these set of CSS cells, and out of these 67 are significantly different for at least one trait. So many CSS cells show like the underlined ones, that means panicle weight is high as well as yield per plant is high. So this is a significant CSS cell which can be used to map panicle weight. Likewise, this CSS cell can be used to map panicle weight, yield per plant and biomass, for which it is significantly different from the recurrent parent variety. So that is how they are used. So in the end, I would just say that we now have a set of 5 CSS cells from 5 crosses in MTU background, in Swarna background, and one in KMR3 background using Nivara and Bufipogon. And they are all in advanced generations. They are stable. These CSS cells are available. Some of these we have deposited with the National Bureau of Plant Genetic Resources, and they can be obtained from Indian Institute of Rice Research. These are the total numbers and how many which are significantly different. And these are the traits: plant height, tiller number, panicle number, total rye matter, for which they are significantly different. And it is interesting that almost 65% of the lines are significantly different for one or the other, at least one trait. These are just maps which show for MTU, BC2F2, BC4F2, for Swarna into Nivara, and we also have for Swarna into Rufipogon also, and similar kind of the variability that we get. The interesting thing was stem diameter also showed a significant positive correlation. So since wild species and cultivars are so different, we get a lot of interesting correlations as well. And here we found that stem diameter was strongly correlated with flag leaf length, yield, and total rye matter, and many other traits. So these are things which we can incorporate in our studies. Do I have some more time?
You can take another 5 minutes.
Okay, yeah, thank you. So these are the founder CSSLs I'm showing here in 37 founder genotypes in Swarna Rufipogon, from which we started. And then we had about 300 in all BC2F4 and also BC4F4. And you can see how many lines are significantly higher values than Swarna. And this is over a 2 generations study, BC2F4, and also we have F5 also now. There are several lines which are significant for more than one trait. So that is the advantage of having this set. We also have some lines where F1 was used as female and another set where F1 was used as male. So we can look at the cytoplasmic effect also in this. There's a good population to work on those things. And so also in Swarna Rufipogon, we found several lines which are low for days to for flowering. That means they are shorter duration and have high 1000 green. The black ones are positive, the red ones are negatively significant. But either way, for basic studies, even the negatively significant are useful because they differ from the variety. These are some of our previous studies which we had done on using different taxations. And we found for 1000 grain weight, we mapped towards 3 QTLs, and we have identified the target CSSLs which we can use for fine mapping. This is one target CSSL which has the different 1000 grain weight and higher yield, and we are using these for other students. I'm retired, but other students are using these for fine mapping. And we found another QTL for tiller number on chromosome 6. So let me just end here now. We also have some applied outcomes from the work, even though developing chromosome segment substitution lines was a basic work. But we one variety was released, Swarna Nivara, for Tamil Nadu, Maharashtra, and West Bengal to replace Jaya. And this is from Swarna into Nivara. And the another variety was for Chinsura Nona Gosaba 2 from KMRC into Orupipogon. And this is doing very well in these areas of 24 Parganas where there is. So we never tested them for salt, but since we had shared these lines with Chinsura, they found that it is doing it has reproductive stage tolerance. So it is better than the saltol containing lines which have only vegetative stage tolerance. And it's giving something like 5.6 tons per hectare under saline area, which is very high. And so these are some kind of benefits that we get out of these lines. There are 2 lines which were very highly resistant to brown plant hopper, and they have been registered with NPPGR. So I just want to say what we started 15 years ago, mapping a QTL on chromosome 2, coming to a sub-QTL, and then finding some genes in underlying the QTL as candidate genes for high yield. It took us 15 years, but I'm hopeful that the younger students who would use these CSSLs can do that now in 2 years. Just to summarize now or give my perspective, these wild parents are not, the phenotype of the wild parents is not necessarily predictive of its genetic value, because these wild accessions were not resistant to brown plant hopper heresy, But when we developed these backcross lines, many of them were resistant to brown plant hopper and high yield, high resistance to pests. Lot of other traits coming. So when we are dealing with wild parents, the phenotype is not necessarily predictive of genetic value. There is tremendous de novo variability which is generated, and Barbara McClintock, the Nobel laureate for the genetics for transposons, she mentioned the word genomic shock, which happens when wild species are crossed cultivars, and that is noticeable. We found a lot of variability generated. The advantage of CSSLs is multiple traits can be improved simultaneously, and it increases our resource use efficiency, and we can share these lines. So, in conclusion, wild relatives of all crops need to be conserved, valued, and used to help meet future challenges of food security and climate resilience. And as the saying goes, if you use them, you lose them. So if you use the wild relatives, that is good. I thank all my students, technical staff, assistant scientists, directors who've been associated, and all the project funding that thankfully for me was continued without any breaks, and the ICR National Professor Project that helped me with this. Thank you very much.
Thank you, Dr. Sar.
I'll be glad to answer any questions.
Any questions?
No, ma'am.
Okay. If there are no questions, I think my duty is to conclude it. Okay?
Okay, ma'am. Okay, ma'am.
Thank you.
Okay.
Thank you so much.
Okay. I would like to congratulate all the three speakers who have spoken and contributing towards the food production, and then what you can just say the livelihood security and health security. Now we know over the past several decades, the use of gems has become widespread in the production of food processing aids and other food ingredients, contributing towards the food security and livelihood security. And coming to Sarala's speech, that is CSSLS, that is the chromosome segment substitution lines. Actually, they are valuable breeding tools for broadening the genetic base of existing cultivars and harnessing the genetic diversity from the wild and distant related species, contributing towards the food security, which is really need of the hour. Now, coming to Dr. Vijayalakshmi's speech, we all know very well the stem cell therapy is being billed as the next pinnacle for all illnesses. The immense potential that has been shown by stem cell in treating of the diseases traditionally considered as a degenerative, incurable, and irreversible, such as diabetes, heart disease, spinal cord injuries, and so many of the other things. Now, I really congratulate all the three speakers for their valuable contribution towards the food security, livelihood security, and health security, which quite often will speak throughout the day. Okay. Now, finally, I would like to thank the organizers for giving me an opportunity to chair this wonderful session, especially Dr. Uma and her team. In spite of all the technical problem, in one side we talk about we are in the advanced stage of in many of the aspects, but many times the advancement may come on the way of hindrance of the work also. But I really appreciate Dr. Uma and her team for meticulous operating and conducting this seminar in a sort of very systematic way with a lot of patience. Congratulations, Uma. And I specifically congratulate Sabita, who is reminding me time to time, because I am busy in other aspects also. Now at 1 o'clock, I have to join the academic council meeting of Acharya Enjiranga Agriculture University. I thank all the organizers for giving me a wonderful session to chair this today's session. Thank you all. And I think if there are any questions, you can have the interaction later on with all the speakers. They are available. Thank you very much. Have a good day.
Thank you, ma'am. Vijaya, ma'am. like your the energy with which you conduct any of the scientific sessions is really great, madam. Like it inspires everyone. It inspires everyone, in fact. And we are blessed to have you here, madam. Thank you so much, ma'am.
Thank you all. Okay. All not today. Okay.
Thank you.
Okay. Saima, it is.
Now for the next three talks. Yes, ma'am. Yes, ma'am.
I got no electricity in my house, so I have come outside and then actually chairing your session.
Oh God.
Power off from the morning 11 o'clock till now it has not come. So I'm using only my mobile internet. Thank you, ma. Sorry. Out this Seva. You just say.
Now it's time to invite Dr. A. Venkateshwari to chair the next three talks of the session. May I now request Dr. Venkateshwari to chair the session? A brief introduction about, one second. A brief introduction about Dr. Venkateshwari. Dr. Venkateshwari is heading the Institute of Genetics and Hospital for Genetic Diseases, Usmania University, Begumpet, Hyderabad. As Director in Charge, which is a premier research service and training institute in South India. She is the Secretary of WST Indian Chapter. She obtained her MSc in 1992 and PhD in 1998 from Department of Genetics, Usmania University, Hyderabad. Her research interests include human genetics, medical genetics, and cytogenetics. She carried out 6 DBT and UGC-sponsored research projects in adult onset disorders, and currently she is the principal investigator of DBT-approved multicentric project on postnatal and prenatal diagnosis of non-syndromic sensory neural hearing loss in the Telangana region of South India, and principal investigator in DBT project on postgraduate diploma program in medical biology, biotechnology, and genetic counseling with special emphasis on prenatal and postnatal diagnostics. 5 research scholars are working under her supervision. She presented more than 80 papers and delivered several invited talks in national and international conferences and visited China, Malaysia, Kuwait, Bangladesh, and USA. She has published more than 100 papers in various peer-reviewed national and international journals and 4 book chapters. Dr. Venkateshwari is coordinator of certificate course in clinical genetics, diagnostics and management, fetal medicine, as well as postgraduate diploma course in genetic counseling offered at Institute. She was awarded International Fellowship for Biomedical Scientists in the year 2016 by ICMR and worked as postdoctoral fellow on anti-cancer role of plant products in cancer lines at Mitchell Cancer Institute, University of South Alabama, USA. She organized seminars and conferences and conducted several health camps at school, college, and community level for general health and genetic problems. She conducted several awareness programs and published articles in newspapers regarding the prevalence of genetic diseases, birth defects, hemoglobinopathies, and cytogenetic. Hello? Am I audible?
Yeah.
She conducted several programs. She is a life member of Indian Society of Human Genetics, Indian Society of Gastroenterology, Organization of Women in Science for the Developing World, and Indian Science Congress. With this introduction, I request Dr. Venkateshwari to chair the sessions.
Okay, ma'am. Good afternoon, everybody. Am I audible to all?
Yes, ma'am.
Am I audible, please?
Yes, ma'am. You are audible.
Good afternoon, everybody. Thank you for your kind introduction, madam. So, as chairperson of the present session, I welcome all the speakers of this session to deliver their significant contributions in the field of biotechnology and bioengineering. It's my pleasure to invite our first speaker, Dr. Kalpana Shastri, former Joint Director, ICAR, NAM, and Consultant, Innovation and Agri-Entrepreneurship Center,
PJT SAU, Hyderabad. Dr. Kalpana Shastri, presently working as professor at Tata Institute of Social Sciences. She is a member of Governing Council, Society for Technology Management, and she has more than 23 years of experience in the areas of technology assessment, IP, and technology transfer. She joined the National Academy of Agricultural Research Management as senior scientist and is currently working as head of the Agricultural Research System Management and Policies Division. She had initiated innovative approaches for new training programs that cover science, law, and regulation, managing research partnerships in consortia in all sectors across the agricultural, supply chain, and rural livelihood systems. Dr. Shastri is the course director of the PG Diploma in Intellectual Property and Technology Management in Agriculture and faculty for middle and senior level management programs like Foundation Course for Agricultural Research Service and others. And she is a member of Indian Council of Agricultural Research team responsible for actualization of IP and technology commercialization policy of ICAR. She has authored more than 90 publications in international and national journals of repute. So, may I now request Dr. Kalpana Shastri to deliver her talk.
Good afternoon, ma'am. Thank you very much. Am I audible? Is it okay?
Yeah. Yes.
Dr. Uma, I have already sent the presentation. Can that be loaded, or do I load it from my end?
Yes, ma'am. It's your wish, ma'am. If you want to present from our side, I will present, ma'am.
Yeah, you can do that. I have no issues.
Okay, ma'am.
Okay, as she starts loading the presentation, I first of all I would like to place on record my sincere thanks to the chairperson who has very benevolently, introduced me. I'm just a normal working scientist, and I used to be in the National Academy of Agricultural Research Management, and I'm by training an agriculture scientist with a training in plant pathology, mycology and plant pathology. So I've been working a lot on microorganisms. But sometime in my mid-career, after about 11 years or 12, I got a chance to be appointed as faculty in the National Academy of Agricultural Research Management in 1996, where I started looking at the option of working into IP. So I do work, I do have a small little experience on IP management and law, and try and help many stakeholders try to, from the technical point of view, create strong portfolios. Yes, it will not be a very, the biotechnological type of a presentation, but I think in all the presentations I was hearing since morning, there has been, a talk of patent, right? In the first one, when Dr. Shashi Rekha had, she had talked about several times some patents. And then later on, we realized when Dr. Vijaya was talking and even Dr. Sarla, that there's so much of research which goes on. And these are the days when there are a lot of opportunities at the policy level, which give, lot of, avenues for researchers like us to actually monetize what we have. Now, I'm not saying monetizing in terms of even money, but sometimes tangible and intangible dividends do come from our outputs, and that's important for us. Let me begin my presentation with a small, sort of a remembrance to Dr. Ananda Mohan Chakravarti. Unfortunately, it was only last week we heard that this one of the greatest microbiologists, the Indian American microbiology, whose simple fight to get a patent in his name when he was working with the GE led to the complete revolutionization of all the type of statutes, the law, the thinking in biotechnology per se, and how this has led to some sort of recognition of biotechnological work which goes on at a very, very great and immense pace. Till that, this judgment, Anand Mohan Chakravarti's judgment, patents, even when Louis Pasteur talked about his role in, it was some sort of a biotechnology, but there was hardly any effort for a legal grant to be given to the output out of the research. And this particular judgment, which was a 5-4, bench, which is just by one vote, completely revolutionized. And I think it's very apt that today's conference in biotechnology, we actually start with his remembering him. Can I have the next? So in my talk now, as per the title given by the organizers, it's on creating intellectual property portfolios through the interference of, I need not really what we call define biotechnology to all the audience here, because we are talking to a leading set of biotechnological researchers, students, faculty. What we are going to talk about is how can we try and build a strong portfolio which can, now why do we need intellectual property? Like in the previous presentations, a lot of papers and all, these are quantifiable indicators for anybody to talk about their research. Similarly, intellectual property these days does give us an opportunity, sort of signal to the outside world what we are doing.
Sorry to disturb.
Next slide.
Disturbance unnadi. Veeru konచెం disturbance వస్తున్నది.
Now,
ఓకే బేసిక్గా ఏంటంటే మేము ఫస్ట్ ఒకసారి చేసిన తర్వాత అడిషనల్ గా మళ్ళీ మేము డెవలప్ చేయాలంటే ఎందుకంటే మీరు అడిషనల్ గా మళ్ళీ ఏమైనా చేయాలంటే చార్జ్ చేస్తారా లేకపోతే ఏమైనా అడిగేది సో దట్ ఇచ్చాను కాబట్టి దట్ యూ కెన్ ఇగ్నోర్ ఫర్ నౌ ఫర్ ద డెవలప్మెంట్ ఆఫ్ ద.
So when we are, so my plan here is to understand the relation between IPR and try and talk about the canvas of IP. Generally, when you talk to any IP means only a patent, but I feel there's a very huge range of work for us researchers, and we'll try to point out at that. Then I'll just go into the checklist, especially for biotechnology patents, because those are a little different and we need to look at this. Now, can I have the next one? And I'll try to talk about certain cases just to, motivate some of our young students who are also part of this. You'll all agree that all of us, even in just look at the previous, starting right in the morning, when the speaker was talking about several microorganisms which they have actually sort of been able to locate, explore, and take it out and then find some use. So those are the assets we are actually base our research on. And we have a huge pool of assets which are of intellectual value. They are value to our research. Some of them, if tuned, can actually be made into a sort of a property which we can say that it is out of my research and I own it. A property, when you call intellectual property, anything which emanates out of the intellectual pursuit of my efforts. I define it like that. I'm not giving even the ready-made textbook definitions, but I thought I should talk in language which can be assimilated by everyone. When we make property, just like when we own home property or land and all, we do have certain rights. And this intellectual property gives you certain rights. Because of those rights, you can tell them that you own it, you have created that intellectual property, but you can also monetize them and thus make them into economic assets. And if you see my entire depiction, the bubble goes smaller and smaller as we walk through. But I can tell you these economic assets are even worth the bigger sphere of intellectual property if properly used. And tomorrow there is, I know, a session on, I think, commercialization and startups. Some of this would be actually directly related to that session also. Next one. Can I have the next? Yeah. So what is the toolbox which I'm talking of? Intellectual property rights are the legal rights. Can you just press on and you'll get all the images? Intellectual property rights are the rights given to the owner through a statute, the legal rights which can be contested. Now, we are talking at the helm of science, it's law. Primarily because we are in a, I mean, this is a little debatable. I've seen through so many years, I've been working in this area for the last 25 years. And every time this fundamental question is, if I have made something, why not give it free to somebody? That's fine. You as a rightful owner are free to give it free also. But you are also in the days where you are trying to look at different business models which are emerging in the markets. And some of our, assets, research outputs which are generated can have a role in this business model. What are the type of intellectual property we are talking about? I go by the doctrine, which is the TRIPS agreement, and India is compliant. No need to go into the entire history. But there are different forms of intellectual property recognized by the World Trade Organization and which have been assimilated in our domestic laws. And today, after, almost more than 96 to this, we are almost more than 15, 20 years away from this, but we have all our rights in place. You have full-fledged offices which are actually functioning towards creating a strong IP climate. You're also in a country where you have, in 2016, the intellectual policy by the government of India, which enables us and most of our universities, institutions, government, public sector, to develop IP policies. The UGC has, for example, under which most of the universities, including JNTUH, is functioning. What are those IPs which are recognized? Copyrights. And I think copyright, if you notice my first slide, the second slide, I would say, in the title, I have actually written, this presentation is copyright, under copyright. A copyright is a type of IP which gives the original expression of anything. It's a tangible one. Or what are copyrights for a researcher? If there was audience, I would have asked it and you would have definitely answered saying research papers. If you, all those publication houses, when they actually accept your papers, the first thing they send you is an agreement. And you agree to give your paper, the paper which you have written on which you have your original copyright, to the publisher who has publishing rights. And next time you want to use that paper, you actually have to take permission. It's your paper, you are the author, you have the moral rights. But if you want to publish it, you need to take the publisher's rights. And this is something I would strongly try to put it across, because you have many young students. You are also part of an e-platform now and a virtual conference. There are a lot of new laws which are coming. Every part of this conference is also under the copyright. And anything which comes out of this is also protected. And the law says the way you write something, the way you pen down type of thoughts, scientific presentation, poems, artistic sculptures, photographs, using the pictures of it in your own presentation, all allowed, and you get a copyright. But attribution to the original author is something which we as R&D personnel never should forget. We should do it. It's very common in many presentations, and I have personally faced this, gone to a conference, and you see your own slide being presented by another presenter without giving an attribution. The take-home message for most of my young students here is learn at a very early age to respect others' copyrights. Copyright is not the right to copy. Copyright is an intellectual property tool which enables and gives rights to the original creator of that articulation. For a biotechnologist, I would say developing algorithms, developing sequences, developing type of genomic what we call sequencing writing, all those which are put into the library. The digital applications in India all are protected under copyrights. I'm not going to the other part of the copyrights, but I thought I'd restrict myself to what a R&D person would really require. The other IPs, of course, very well known are trademarks, patents, of course, I will talk about it in much more detail, designs, geographical indications, which perhaps will not be part of a portfolio for a biotechnologist, but at the same time, we must be aware that we have several community-owned intellectual properties which are already known for centuries, which can be, if by the law, if stipulated, protected under GI. But we will not talk about all this at this point because I would again reiterate. Can I have the next slide? Now, if this is the toolbox, and I said there are laws already in place. Unfortunately, for biotechnologists, you must remember we are talking about something which is already existing in nature. Ananda Chakravarti's case itself was a symbolic and contested because everything occurs in nature. So you discover microorganisms, you discover the genes which are giving this. But then when you find some value to it, a human intervention comes, and it's that small space which affords. So you have the Patent Act and all these acts which are forming in one circle. You also are having in a country where you do not give patents to plants, which Dr. Sarla was talking about, improved crop varieties. You register them under the Protection of Plant Variety and Farmers Act, Rights Act, primarily because we feel registration is in IP language, it's not so strong a portfolio as a patent is. You also have the Biodiversity Act, which actually recognizes the sovereign right of every citizen on any material which is a biological resource. So it's everyone's. Now, how do we, when these are the overlapping mechanisms, can I have the next one? I have tried, next one. I have tried to put all these into one table. I know it's a very noisy slide and a crowded one, but I thought it's easier for us to discuss. So if you're talking about patent, the Indian legislation is the Patents Act 1970, which is the administrative authority, which is the Controller General of the Patents, Designs and Trademark. And what are you trying to look at? Anything which is a tangible process, product, which is novel, non-obvious, and industrially applicable. Only three attributes, very important one, as we discuss further even. And this is something which is attractive to several biotechnological-based researchers. And patent is definitely a tool which is more viable. And design is looking at, under the Design Act, it's an intellectual property which is been after the revision after the TRIPS agreement, which gives us for any original depiction in the 2 or 3 dimensions. So you have say a health drink which is based on a gene expression which can be patented by a process product, but you want to put it into, say, a bottle which has got a straw and which is without even putting your hand, you try to drink through it. It's a new design, a two-dimensional, three-dimensional, which affords more aesthetic, which is distinguishable, which is also original in thinking. So design is one more. A trademark. We are nearing lunch and we've had a heavy session since morning, all of us. And some of our students, if you get frustrated, of course, we can't see you, but then if you were to say you have a headache or something, the first thing you would do is to go and pop up a Saridon or analgesic. You would never say, I would go for a paracetamol, or you want to go to the normal, go and buy something. You just go on to the Amazon online. Why do you go to Amazon? Why don't you go to the other trade? There, it is the brand which is speaking, the trademark which is in your mind. And if you want to take your research outputs into a more better business model, branding is something which is of much value. The rains have come in, and since I'm agriculture, I have a tendency of giving examples from ag. When you talk about farmers, going in brine, they just try to buy those seeds which they are more comfortable, primarily because it is from a company. You want to go and buy a toothpaste, you'll never say, give me a toothpaste, you say, give me Colgate. And Colgate is the trademark. So when we develop biotechnological based devices or tools, many companies actually brand them for business through trademark. Copyright I've already talked about. I have deliberately put the ones which are pertinent to biotechnology in a different color. So plant varieties, you can have a patent on a gene, construct, or a primer. You can now have. But if you put it into a variety, you can register under the Plant Variety Act with the Protection of Plant Varieties and Farmers' Rights Act. Can I have a registry? Can I have the next slide? Before we go into further, I just want, because it is on biotechnology, there have been important case laws at the global level also, which have been very, very helpful in developing the evolution towards treating biological material-based inventions all in the used for developing. The next, but more important, is the last case, because that was one. Next one, that was a case in India which actually, can I have the next one? Which actually in 2002 reversed a judgment where a living material as part of a vaccine production was granted a patent. That was the first case. I mean, yes, 2002 to 2020, it's a long journey. And today, many of us are very comfortable because many of our, interventions through biotechnologies are ready in the court of law for a grant of a patent. But the history has been pretty evolutionary in respect to biotechnological research. Next one. Which I just wanted to, motivate some of you people. So any method we are now into the genomic area. You have CRISPR-based genome technologies coming in the healthcare, in agriculture, in food, in any industrial biotechnology. All these are now finding a place in the patent office, and there are immense opportunities for developing a strong portfolio for us. Next one. Where is the next slide, please? Now, before we actually do, okay, you are having all the toolkit, you are having tremendous amount of opportunities. But I particularly put this slide only to motivate some of the young students who are there. A good combination of science or biotechnologists with legal knowledge, and also an attribute in able to do informatics is a huge opportunity of career. There is a dire shortage of such qualified professionals who have a strong background in biotech, working background and working knowledge in biotechnology, in the legal parameters, as well as developing an acumen for understanding the patent knowledge itself. So that's called IP informatics. It used to be mostly patent informatics, because I'm sure many of you are owners, some of our faculty and speakers I know have a lot of patents also, and the patents in biotechnology run into hundreds of pages. So if I want to decipher as a source of information, just as we see papers, there's a new trend which comes, new sub-school which is developing at the global level. It's called IP informatics, particularly PAT informatics, which combines the skills and gives a lot of opportunities, of course, jobs. Also, it's exciting because last 3 days I've been actually evaluation for DBT. At the time of grant of projects, post-grant, at the time of, IP is asked the possibility of the IP, as well as what is the level of prior art search you have done before you put your proposal. So any grant, this dimension is important. And prior art search, informatics search, ability to understand and decipher is equally important. This is a recent paper from our team which we have. And we realized that CRISPR technology, which is into the health care sector much more, is also made into the agri sector. And very useful information. If I were to give funds for a CRISPR technology based proposal, I would need to know what is the white space available. And you can actually understand, because no donor likes to fund projects if at the end of the game, his product is already in the market. You are looking at novel products out of research grants. And informatics is one tool we must all try to build our skills based on what is known in the IPA. Next one. So if I were to, as a researcher, it's not easy, let me be blunt, it's not that easy to get a patent. It's also not difficult to get a patent which is based on biotechnology. We must be actually knowing the checklist. Next one. And so I just tried to put all the checklists which will be. All biotechnological inventions for a patent mostly are patentable if they are novel. That means nowhere in the world such a invention or process product has been actually been known. Means it is not published. There's no prior art to it. There is an inventive step to it, which means even though it's novel, it is a step. Suppose, for example, I'm trying to one process which takes 5 steps. But because of my intuition, I bring it down to 3 steps. This inventiveness of the nature of the invention has not been recognized or deduced by any other person of equal caliber in this field. That forms an inventive step. Industrial application, even if the patent is granted, anybody sitting across the world can view it, can understand the steps, and is ready to take this for technology upscaling. That means you need not set up industry as a owner, but you can license your technology. It should have a repeatability. And it should also be as per the law of the land. In India, we have a section, which I won't have time to do, the section 3 and the section 2 of the Indian Patent Act, define certain inventions, even if they are novel, inventive, or industrially applicable, they are not patentable as per the law of the land. So when we are trying to patent any invention, we do go in and check section 3A to section 3H. For example, no patent which is based on traditional knowledge is patentable in India. No patent which is as per section 3H, which is agriculture, is not. That means any traditional type of plow or any variety which is already existing is not patentable, even if you say it's novel and inventable. And patentable, the checklist is there has to be a written disclosure, special disclosure requirement as per the law, and a deposit of the material on which your invention is based at the identified repository. Next one. And the next slide, please. So there is a treaty called the Budapest Treaty, on which the IDA, you have, of course, in Chandigarh, the only authority, the IDA, which is located in India for some of the type of materials is at Chandigarh, the MTech. But you also have other 23 plus institutes all over the world where you have to deposit this material, your material in a prescribed form, and then attach that thing when you approach the patent office for applying for your patent. Next one. So as I said, the MTC and all types of the GMOs, the bacteria, the viruses, the cell lines, seeds. Seeds, of course, we deposit in the Plant Variety Act under the for the plant variety protection, plasmids, and any other, even DNA, mRNA, all these. Why do we deposit? We are having a patent grant for 20 years. So we are trying to have the basic material on which, with a chance for accessibility of that culture to other people who will base their research on this, after giving you acknowledgement and go further. So this is a mandatory compulsion under the Indian Patent Act. Next one. As now talking, and this I think under the section 10, the special disclosure requirement. I'm sure many of the scientists will say, anyway, my lawyer will know and my office will be approaching the patent agent to do. But let me tell you out of experience, it's important as a researcher to at least know the fundamentals so that we know what we are writing. You are the owner and you should know what is disclosed. Next one. And these are some of the other details which you have to give, including the permission from National Biodiversity Authority, which is based in Chennai. It is mandatory that any material which is based on any biological resource, you get a permission from the National Biological Authority or the State Biodiversity Board in all the states. There is it's a normal procedure and it is time bound. So many of the applicants do have fears of getting permissions on time. You need not really bother. You will have all these things. In case of sequence listing, it has to be there. The listing has to be given in an electronic form also. Next one. Now, this is the checklist, and which is a very office type of work which I am doing. Till now, we have tried to understand there is IP law in place. There are kits of IPs which you can make. I will try to sensitize some of you people to discussing certain real-time cases of which we have in the past. So we'll have the next one. The first one, I'm just taking cue from Dr. Sarla's presentation, and this is the case of Samba Masuri, which is very popular in the region we are talking. Samba Masuri is something which all of us rice eaters are very familiar with it. It is one of the varieties which has been developed way back in 1986, and still we call it agriculture rural use. time. But if I have a capability and trying to understand to induce the resistance to bacterial blight, which is taking a toll in many of the rice fields, I can use the biotechnology platform. And if I induce this, this is an act of human intervention using human intelligence, which definitely warrants recognition through, IP. Next one. So a team of researchers and my colleagues from IIR Rice Research Institute, along with CCMB, had a project together, and which actually led to the development of bacterial leaf blight resistant derivative of Samba mussel. So what type of IP you can? It was it's not a transgenic, it's a non-transgenic approach, the product, which can be liable for a strong patent. Also, the variety which has come out of this entire testing is now registered under the PVP registration with the BPF product. So you have two already IPs in place. You have licensed this technology for further production of this seed companies through a license agreement. So you are not taking it forward, but those two agencies, the two companies, can brand it in their trademark, so you get a trademark also in the portfolio. And it's a typical example of a public-public organization bringing into a change which reaches society. In fact, this particular variety is also very well known, not only for the high yield, it is also low GI. So in these days of health consciousness, it is doing very well in terms of the GI, glycemic index lowering. So it is very popular with some of for health reasons. Also, this is one variety which stood very well in the field during the Hudhud cyclone. That means it was able to withstand, while the traditional Samba mussel, the ISM withstood. So with all these probabilities, it definitely was worth going for licensing. Next one. Next. So what I was trying to say, bring out to is strong IP portfolios can be built within the research groups also. That's important for us to understand. And it can be where science can reach society in a planned manner, getting acceptance, getting good market, and also increasing the income for everybody and keeping the consumers happy. Next one. I'll now take you into something which is very not Indian, but it was interesting to me. I think everybody knows about cranberry. In Telugu, if I am Vakkai is something we call it. It's cranberry or karunja, which is called in Hindi. Next one. It's interesting to see that a type of a crop, a small, which is not very popular, and it's a crop which is not even grown well. And next one, you can quickly go through this. It's okay, I can explain it. Cranberry is off late. If you go to market, you will see it's a fashion among most of our youngsters to say that they drink cranberry juice, which is available in many of our supermarkets. It's also a very good source of vitamin C. But wild cranberries are something which is not a great crop anybody does work. By what we call a device to harvest it much easier, and which was this principle was used to make an automatic sorter of the fruit. Now, next one. If you look at world market, ocean spray is next one. So what happened is there were cooperatives which was created. People started growing. Yeah, let's stop here. And cranberry was there. Cranberry is something even if you use for dal making, you use 2 or 3 fruits. It's not, and at the most you can use. But when you have so much of produce, you also have to think of, I mean, innovation comes in and people start thinking about new ways. There was one patent which actually had a little bit of invention where the hull was reversed. The process hull was filled with other fruit juice. So you started to have flavored fruit pieces. And the surplus production of cranberries, which was not being able to use anywhere else, led to one small product, which is part of a gelatin treated, fruit pieces, which were tasted with orange and cherry. So there were two uses for this. One, you could have these small fruit pieces, which can be part of many of the bakery products. And the same source of cranberry in shortage of orange juices, the company started using cranberry juice as a source of vitamin C for them to put. So one patent, then of course they had the gelatin treated food pieces also. Next one. And all your breakfast cereal which you're seeing today in the market, next one, is actually, can I have the next slide please? All those, small cherry pieces which you are seeing in bakery products, most of them, can I have the next slide please? Hello? Can I have the next slide please? Yeah. So we started this particular patent, which was patented in many countries, led to those small red and white, pieces which you are seeing in many of your bakery stores. It is not a great product by itself, but the patent has fetched and built a whole business of cranberry, a poor plant which is ignored, which is a waste, which now forms a complete source in the food industry. Next one. And the craisins is, if you go to the US, craisins is part of the several food, breakfast beverages and breakfast cereals also. What I'm trying to say is you need not have high-tech also. A small invention can actually build a strong portfolio of Ocean Spray's trademark, of a patent which they have a valid patent for 20 years across the different nations in the country, and they're able to make huge money. And this is owned by a cooperative. It is not owned by an inventor himself. Next one. I will now talk about the last case. Oh, okay. I have also put value case. Value is in a country like Finland, milk is something which is very popular and advised, I think everybody. But lactose intolerance, and I'm sure my friends from NIN will agree, perhaps not a big problem in India, but emerging today. Lactose intolerance is more there in Finland. This particular Finnish company, licensed dairy products, had a patent which would help make a lactose intolerant milk. And today, this product, with a good, strong trademark, with a patent, with everything, has entered the dairy product industry with a very strong, all its strong IPs, and it rules the world. Next one, you can quickly go through because we are talking about food. I feel rice, we talked about cranberries, we are talking about milk, and that too just before lunch. I think it is definitely an area which will motivate. What I'm trying to say, bring here is an intervention of trying to produce lactose-free milk, which is patented, has helped to bring, and which was the consumers needed, less sweet milk, because that consumer wants that light milk drink, and it actually fetched. This company survives only on this. So an IP which is developed, which can actually make big companies also survive on small invention, is the message I wanted to tell. And the next fruit, you had milk, you had rice, you had berries. Next one is kiwi. Next one. We can skip all this. It's okay. I've already talked about it. The kiwi fruit is something, again, you will all agree that we didn't grow up with kiwi. Okay, we all see now kiwi as a very elite fruit. And kiwi is actually a fruit which is local to China. It was brought from China to New Zealand. It made its field there and led. The kiwi fruit has come only because it was grown in China, in New Zealand, but it's original from China. It's an interesting case. Next one. And when we want to buy kiwi fruits today, you can skip this, you can just skip. You will realize that this crop is the economy of a country like New Zealand. They really have spent a lot of R&D in a PPP mode, public sector and private, to have a very strong IP on it. And today when you buy kiwi fruit, even from Spencer's, you will see there is a small little label attached to it. It's a trademark of the company, which is the main company of kiwi fruit, which is also coming into Indian market. Next one. They found that the wild varieties and processing, they have almost total number of patents on this particular kiwi fruit, and the processing part is almost, it's a huge portfolio. And you have so many companies working in it. In fact, even CSIR holds a patent in kiwi processing. And they also have strong trademark. You would agree, if I were to ask you, just a company which is you're buying the kiwi fruit, you would go for Jasper. Next one. And you'll see that company's logo is always there. This has been because the type of range of kiwi fruits, sweet to non-sweet, yellow, green, antioxidant, all this with the public sector, institute. Jasperi has partnered in a huge breeding program. They have protected them with trademarks, with plant variety registration, with an IP on the process and a patent on this process. And they have delivered a variety of tastes, colors, and sizes to their consumers across the world. And this has actually happened. Today, what you're seeing is a delivery of all these IPs only. Next one. So what am I trying to tell you? If you want to get into the market, and in the session tomorrow, you have startups, you have young researchers who have small inventions, but they can make it big. IP enablement is only helping you to go. It's not that you will, there's some negative feeling of an intellectual property that it's only money. It will raise costs, but it will also help get recognition of the efforts you are making. It will also help you to license it to people who really need it. In case of biotechnologies, there are several types of technologies, I forgot to actually tell, which are protected under trade secret. See, those are culmination of certain steps which are essential in your process, but they are not leading to a product. Such things are protected under trade secrets. And tech transfer offices in any university are responsible to see that to give it to others on a trade, also on a trade secret. Everybody knows a classical example of trade secret is Coca-Cola. You are drinking Coca-Cola for years. It's a product, years it is there. It has a design on the type of bottle. But no one knows the formula of Coca-Cola. You know it generically, but you can't know the specific thing because it's a protected one. And the entire business runs only on a trade secret. So biotechnology, you do have several processes which can be protected under confidentiality agreement, which is the way instrument in India to do it. And all your offices do. So I hope in the time which is allotted to me, there's a lot to share. There's a lot of trigger in IP which is, and I would like to cut it short here by saying that appealing to some of the students who are looking for new inventions, carry on with your invention. There is a lot of scope for you to build IP portfolios. Think of licensing and trying to go forward. If you're not interested in, making a remuneration of it, license it and continue your research. If you are planning to go, you can have startups, you can have your enterprises, and the government, the current scenario, the type of scenario which is there in the country today, there will be a lot of opportunities for you to actually get mentored and get into starting your startups. Biotech startups, if I were to start talking about CRISPR, the two big startups which have revolutionized CRISPR into agriculture are actually fallout of two university technologies generated from students. So it can happen, and the field is huge and open for everybody to trigger innovation and entrepreneurship with recognition through IP portfolios. Thank you.
Thank you, ma'am.
Ma'am,
Thank you very much for your
time, ma'am.
Hi, Kalpana, can I ask questions now?
Oh, sure, ma'am.
Okay.
Nice to see you.
Hi, yeah. Considering that there must be a lot of students over there who done molecular biology, wanting to become entrepreneurs, and many people tell them, like, for example, Shanta Biotech is a living example for most of them. So then they turn around and ask, is that a process patent? Yes and no. Then you say, I mean, PKR was existing, so it's a discovery. Then could you please clarify to them from your language that the construct that you're making itself is innovation?
It is not discoveries are not patented.
So it's not a discovery. That's what I'm trying to tell you. Making a construct is innovative. Two, and then making that construct into a possible process for a beneficial product could be a process patent. So could you tell them, like, how every construct that a child makes at the university could be licensed out to become a good process, and they make a licensing fee out of it? Can you explain it to them? I might know it, but not being a patent person cannot explain that. Thanks, Kalpana.
Constructs earlier, prior to 2000, 2002, were not allowed in India. Now, yes, it's a big yes. Having said that, let me also bring it to this. There is nothing like a process patent and product patent. After the amendment which happened in right up to 2002, the amendments happened in our because we had to be compliant with TRIPS agreement. So anything which is falling tangibly in one go, it is novel, it is inventive, and industrially applicable. All three together. All three together are defined for a patent. Having said that, if the construct, you would have to also give the disclosure. If it is based on certain natural resource, biological resource as defined by the NDA, you have to take, that's very fast, a permission, and actually it's an information to NDA, and then apply. These you have to do. But things are really good because I go into the patent office also as a scientific advisor, and I know the pace at which I can say there's a huge amount of difference in the way things are moving in India now compared to, say, 20 years back. It is mind-blowing. And I actually, even in COVID time, I know we've had hearings online. So things are moving. I started my work in IP where everybody was saying, oh, in biotech, nothing will happen. I don't want to raise the news video case, which is in the court and all. Yes, you must also remember, in case of biotechnology, the type of knowledge, information, the evolutionary as science is also at a very rapid pace. And the law has to also be in this pace. So now the younger lot who are examiners are very compliant. They really know the subject also. It's just that when we were doing this CRISPR paper, it's a very recent one, there were so many, holes in it, because people also are not able to interpret even at
Ma'am. Thank you.
I have answered construct, yes. It's a big yes if the three are in case. Yeah. Geeta, anything else?
Already we are shortage of time, ma'am. Thank you. Thank you very much for your lucid presentation on the importance of intellectual property rights in the field of science and technology. And also, we are very much enlightened with your sharing of your experiences and case studies, so that we could able to get the information regarding the Indian scenario at present. So thank you very much, ma'am. And due to shortage of time, questions will be posted in the chat box. So please answer them. Thank you, ma'am.
Thank you.
Thank you. So the next speaker of this session is our convener of the conference.
Madam, 1 second. Dr. Saida is waiting, madam. So let Dr. Saida finish first.
Okay. Okay. So we move on to the next speaker, Dr. Ravi Saida, Assistant Professor and Co-Convener of the conference. And Dr. Saida is presently working as Assistant Professor at Center for Biotechnology, IST, JNTUH. He completed his BTech Chemical Engineering from Osmania University, MTech Biotechnology from Anna University, and achieved PhD under the supervision of Professor M. Lakshmi Narasu and Dr. H.S. Oberoi. He started his career in the Department of Biotechnology as Assistant Professor since 2006, and then he was given additional administrative positions like Additional Controller of Examination, worked in the confidential teams for various government exams conducted by JNTUH on behalf of Government of Telangana and Andhra Pradesh. And then he was a chairperson for Biosafety Committee, member for Lonza India Private Limited, BOS in the Department of Chemical Engineering, College of Engineering, and PTPG Coordinator, IST, Head Center for Biotechnology, Nodal Officer for Techie Program of JNTUH, Hostel Manager, IST, and Purchase Committee Expert Member, Biotechnology, and University Library Purchase Committee Member, and also he is member of Admission Results Processing Committee. The 12 years of his teaching experience in the Department of Biotechnology and research experience in collaboration with different organizations like Indian Immunologicals, Hetero Drugs, Biotechnology, and CIPHET, Punjab, and others in different areas related to biotechnology and chemical engineering, handling 2 projects presently had made him get bound to the biotechnology department in depth. Under his supervision, 50 MTech students completed their projects and presently guiding 1 full-time PhD student. Nearly 15 publications were published in reputed international and national journals, and has played a convener role in the international conference organized by Center for Biotechnology. Apart from that, given many keynote lectures and invited lectures in various national and international conferences. He had attended many training programs at various universities like Windsor University, Canada, Kansas State University, USA, IIM Kozhikode, and pedagogical training, NITTT and Research Chandigarh, NIT Warangal, IIT Madras, BIOS in Bangalore, Indian Institute of Chemical Technology, Hyderabad. May I now request Dr. Saida to present his talk.
Thank you, madam, for your elaborate introduction regarding me. And I would like to thank you, the convener of the conference, Dr. Uma madam, for giving this opportunity to deliver talk for this session. And I'm going to deliver the talk regarding the media optimization technique for the NM production. So here, basically, so this type of technique is going to be useful for the scholars as well as for the MTech student and MSc students. So hopefully I would like to request all the students, please take this talk for your the research application. And hopefully I would like to finish as early as possible. I don't want to take much time. And here,
Can you share your PPT?
I'm sharing, madam. It's not. Hello?
It's not visible. Saida, it's not visible.
It's not visible, huh?
It's not visible, Saida.
1 minute, one wait. Share my screen. Now, okay, madam?
It's appearing.
Okay?
It is, yeah, coming.
Right. Right. So here basically I'm going to discuss the media optimization technique, that is RSM, the response surface methodology. Basically this RSM is going to be a mathematical, a statistical techniques, but is going to be useful for the designing of any process parameter. And a process parameter, sometimes we are going to be optimize the physical process parameter, or sometimes we are going to optimize, okay, the chemical process parameter. Sometimes combination of the physical and chemical also. So this technique is widely useful for the pharmacy students, biotech students, chemical engineering, mechanical engineers. So basically, so best and most comprehensive. So I have taken this RSM as given by so Myers and Montegrid. This is major sources, and the software also it is there for regarding the RSM. That is the design expert software. 6 initially started, 7, and as well as now it has improved. The improved version also there. So you can able to see website also. So www.startAce.com, and so we can able to use how this software also you can able to handle. Generally, this RSM widely used for, I was telling, the product development, and objective of RSM is what here I was telling, the mathematically and the statistical techniques useful for modeling and analysis of application, where the response is going to be, where the response is going to be, just I would like to cover. Where the response, we are going to see that in terms of sometimes of how many response we are going to consider, sometimes depend upon the process, we are going to fix the responses. So how this response we are going to optimize. So with help of RSM, we can be able to see that. And here sometimes the response so sometimes the response may be maximize that response, or sometimes we are going to minimize that response. Sometimes we are going to get the target of the response. So basically, RSM is objective of to achieve the response in form of a maximize of that response, or the minimize of the response, or getting a target of response. So here basically a non-linear model is warranted when there is significant curvature if the response is going to be happen. So now you can see that the concept of the response surface model. So we are going to give the input, and as well as so block box system is going to be worked out, and we are going to get the responses Y. So here initially the original system is to have that unorganized fashion is to be there, the different parameters. So with help of the DOE and with help of RSM, the DOE means design of experiments. So with help of that, we are going to get a proper organized of the model that's called a response surface model with help of a quadratic surface model. So here you can able to see that this a proper organized model we got it. Y equal to beta 0 plus summation of beta I xi, or summation of beta I xi square plus summation of beta I xi xz. So this is the basically the quadratic mathematical the model to come to work out this response surface model. Here in this the response surface model, we are going to discuss two models. One is the plucked worm design. Second is the central compound design. So entire for this, for example, let us see that. So in a media, we have the different composition, that's carbon sources, nitrogen sources, trace elements, growth factors, etc. So all these things we can able to first investigate it. Basically, traditional methods, which are the carbon source is sensitive to our production or the process, as well as same to which are the nitrogen sources. First we have the idea, but different carbon source we have that, different nitrogen source we have that, different phosphorus source we have that, sulfate source we have that, trace element we have that. We have to screen. So first of all, we are going to use the plucked element design. After screening, we are going to get it what here. So which is a important the parameter which are the compound composition. With that composition, we are going to be optimized. That is, I would like to say that example of a campus interview is to come to company. Company is to come to the campus placements. First, they are going to have the screening method, that is screening test. After screening, they are going to select it. They are going to give training, that is called we can say optimization. That's called CCD. So screening part is going to take place the plucked Berman, and as well as the central compound design is going to take optimization. That is only the two methods I would like to enter first. So here the plucked Berman design is what here? The statistical techniques, the widely employed for the screening of the major constituent of the media. This model, the plucked Berman design doesn't describe interaction among the individual variables, as well as so this plucked Berman design So it's going to start minimum 7 variables, and the number of exponents should be multiples of 4. Mean what I can say the 8 as well as 4 to 8, 4 to 12, 4 for 16, like that the multiples of 4, and followed by always we have the number of variables. We have taken 8 variable, what here 8 minus 1. The 7 variable we have to consider. Sometimes we don't have 7 variable, sometimes we have only 5 variable. So to make up the 7 variable, we have to add the dummy variable. Let us see the 4 3 are 12, but we have to have the variable. How many variable you have? Should you have that? To conduct the experiments, multiples of 4, 12 experiments, but we should have the variables are 11. But we don't have 11 variable, we may have the 9 variable, or we may have the 8 variable. But in order to make multiples of 4, then 3 dummy variable. Mean what here? 3 dummy variable we are going to add it, because so here 12 minus 1 11. So in order to make 11, 8 plus 3 dummy variable we have to add it. That is only this statement has said that if the variable is less than 7, then we should introduce the dummy variable. It means which variable doesn't show any effect. Let us see that, for example, a certain process, let us see agitation doesn't show any effect in the response, or enzyme production, or enzyme activity, or conversion. It could be anything. So that time we can say the RPM is what here? One of the dummy variable. Or certain experiments what here? You can say that what it mean. So like moisture content also, the process may not be important, or particle size. It could be anything. Which parameter doesn't show any effect on the response, that variable we can consider the dummy variable. So here, to design the pluggable designs, we are going to have, the coded values, that is H is a high value of that variable, L is the low variable. So we are going to design, for example, I was telling we have taken the 7 variable, A, B, C, D, E, F, G, 7 variable. Out of the 7 variable, I would like to consider how many experiments we have to conduct upon the pluggable. 7 plus 1, 8 experiments we have to conduct it. 4 to the 8 multiples of 4. So here sometimes let us see, I would consider E. So in this experiment only I have the 5 variable or active variable. So in order to make up the 7 variable, I am going to consider take E and G are the dummy variable. So with that, according to pluggable design, we are going to design the experiment. First experiment, A value should be H value high value, B value high value, C value high value, D value low value, so E value H value. Like that we are going to have, so first experiment, and we are going to conduct the experiment, and we are going to get the result is, so we are this result what here, we are going to consider yield. So that is 1.1. Like that, the second, third up to 8th variable. So here one logic is what here, how we are going to design this pluggable design. Only simple logic what here, each variable should be conducted equal number of time high level, equal number of time low level. Let us see one. So A variable 1, 2, 3, 4. 4 times it has taken high variable, high level, and 1, 2, 3, 4 low level. So like that, if you see that any variable same logic is going to follow. So equal times of high level, equal times of low level. But how we are going to consider here, simply logic is what here, H, 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 LH, LH. So first L you take here and push the first experiment, then you will get this L. First L you take once again third experiment, then push the second experiment, then you will get this thing. Like fourth experiment like that up to, so only what here, 8th experiment we are going to conduct only L only. So if this follow fashion up to 7th experiment to pushing up the previous experiments, so start with the last variable, then you will get this, the chart, and followed by 8th we are going to conduct LL. So there is no hard and fast rule that only which of the logic you follow, but each variables either C or D or E. So equal number of high level, equal number here 4 times high level, 4 times low level. That is why logic is going to follow. Next, here after conducting the experiments, so we have to calculate the yield value by experimental data. Then we have to do the first point what here. So which is the variable is important variable with the help of F test value. For that, we are going to follow the 4th step. One is the effect of variable you are going to calculate. The mean square of the variable you are going to calculate. That is called a variance effect of the variable you are going to calculate. The mean square error you are going to calculate. And 4th step is F test. So that is the F test nothing but here mean square of the each variable by error mean square. So 4th step we are going to give the value. So previous my research what here, I have taken A, B, C, D. So here D value I have already taken what here. So D value dummy variable. So A value is we got F value is 46.4. F value this is, and B value this is. So highest F value is the important variable. Next ranking is B variable. Next ranking is F variable. Next last ranking C variable. D is the nil effect. So like that. So this is a formula how we are going to calculate the effect of each variable. 2 into summation of A variable high level and A variable low level by 8. And the variance effect how we are going to calculate. So like that, effect of variable A, effect of variable B, C, D, E, F, G. we have to follow. Same thing, variance effect also we have to calculate with the help of this formula, summation of AH minus summation of AL. Mean AH mean what here? So I would like to consider AH mean what here? A. So what is the yield value? 1.1 plus it's 0.8 plus 0.9 plus 1.1. That is we are going to calculate it. So here, like that we are going to calculate, then we are going to get this effect of variable A value, same like up to, so G variable, and variance effect also we have to calculate with this formula, and up to G variable, then mean square error we are going to get it. That is the mean square effect of variable dummy variable E plus mean square effect of variable dummy variable G by 2. That's called mean square error. And F test value we are going to calculate not only F test value of B, C, like that. So with the help of this formula, the mean square effect of variable A by mean square error effect. Same thing, F test of B mean what here? Mean square effect of variable B by mean square error. Next, F test value of C mean the mean square effect of variable C by mean square error. Like that, this formula basically we are going to follow it and we are going to calculate it. So the variable A, B, F shows the large effect which are very significant, where C shows very low effect which is not significant, and D shows no effect. So A, B, and F have been identified as the most important factors. So like that, so according to plug-in chart, we have the software, design software will show that in my cellulose production I have taken, so media composition, potassium diphosphate and calcium chloride and dipotassium phosphate and ammonia diammonium sulfate, and these all in trace elements I have taken, urea I have taken. So in that what here, so here 3 factors has shown the positive effect, the remaining, so here the 4 factors has shown the negative effects. So my intention is what here, so in media composition these 4 factors I will keep as such only, and remaining the 4 factor I would like to optimize from the low level to high level. That is only I am going to do next step, that is called central composite design. Here, so media optimization would be done by the, I was telling, so with the help of the plug-in design by screening, next we are going to do optimization with the help of the CCD. So here the CCD we have that, but box banking also people used to use, but mostly people used to use in RSM the CCD. Here the CCD mean what is central composite design. So the central composite design is going to be helpful for you. Here not only low level, high level, the CCD is going to be study investigated up to 5 level: low level, high level, extreme low level, extreme high level, and optimal level. So that is, so what I can say what here, 5 level it's going to be study that, so which level is going to get the maximum response. So that is going to be happen here. So I was telling here, first of all, what here the coded language is going to be used a different way. So the coded language what here, -1 is the low level, +1 is high level, and 0 is what here, optimal level, that is 3 level. Earlier the plug-in has used the coded language L and H, but here -1 is a low level in CCD, +1 is a high level, and as well as 0 was the center level, and -alpha is extreme low level, and +alpha is a extreme high level. So this way we are going to see that how, so in X1, X2, X3 factors, how we are going to consider next year. I was telling, so first of all, each variable, let us see that A, B, C variable. A variable has a low level, high level. B variable also has low level, high level. And C variable also has a low level, high level. If you make it 2 variable combination, that it has this level of studies. So 3 dimensions we can see that. Next AC like that, BC like that. And if you combine it A, B, C, these studies we are going to get. That's why according to CCD, we are going to study the main factors, individual variable effects, 2-factor interaction, this is, and 3-factor interaction. That is the things you are going to study in the CCD. So here the CCD is going to consider, I was telling, this is the center point. Each variable, let us see that X1, X2, X3. X1 center point this is, and X2 center point this is, and X3 center point this is. And what I would like to say here, X1, this is what here. So let us see that this is cardinal point. This is cardinal point. How many cardinal points we have the cubically? 1, 2, 3, 4, 5, 6, 7, 8 cardinal points we have that. And center point, how many center points you have that? The 4 center point you have that. So here one, here each variable will go low level to optimal level, center point, and up to center point to the high level for X1. X2 what here once again? So here low level to center point, and the center point to the high level. Next what here? See that the color also once again. So low level to center level to low level, and center level to high level. So this is we have here 8 corner points. That's called a factorial points. 1, 2, 3, 4, 5, 6, 7, 8. So I was telling 1, 2, 3, 4, 5, 6, 7. So this is the factorial point, nothing but a corner points. Next one here, axial points. Axial point I was telling one extreme, low level, 4 level we have that. 1, 2, 3, 4, 5, 6 level we have that. So this is C. So here, extreme low level, extreme high level. Extreme low level, extreme high. So here 8 corner points and 6 axial points, and here the center points. So this is center point we're going to consider here. So the center point we can consider here the 3 levels, because x1 one center point, x2 is the center point, x3 is the center point. So like that we are going to see that. So here CCD is going to be study. I was telling the 5 levels, and this can be done by factorial points. That's called a corner point, 6 center points, and 3 axial points. So this is the CCD concept. So CCD, I mean, how many number of experiments we are going to conduct? So the total number of experiments to be conducted, 2 power k plus 2k plus n0. K mean number of factors. So let us see that I would like to consider 3 variables, 2 power 3, 8 variables, plus 2 into 3, 6. So 8 plus 6, 14. And we are going to consider 3 times center points. So 14 plus 3, okay, 8 plus 6, I mean 14, 14 plus 3, 17. If you are going to consider 4 center points, then 18. So like that we are going to have the formula of number of experiments to be conducted in CCD. So this is how we are going to design. So this is the corner point, this is the axial point, this is the center point, right? So here we have, I mean, how we are going to calculate this. We know that what here we are going to low level, high level. We know that the center point what here low level, high level average is the center point. That's called a 0, 0. And we have to know that important point is how we are going to calculate extreme low level value and extreme high level value. That we'll see that next step. So here I have taken in media composition these 3 parameters are important in the cellulose production, that is the glucose and diammonium sulfate and dihydrogen phosphate is the 3 things is important. And here the low value is I would like to consider the glucose is 5 gram per liter, high value what here 15 gram per liter. So 5 plus 15 what here 20, 20 by 2, average is the center point is 10. Like that here 2 gram per liter for diammonium sulfate and 4 gram per liter for the, okay now. So diammonium sulfate high value. So 2 plus 4, 6. The center point 0 level is the C 3. Like that 1.5 is a low level, 2.5 is a higher level. In what here the 4 by 2 is 2, 2 is the center level. So this is the calculation of the coded. This is what here -1, 0, +1, plus alpha, minus alpha is called the coded value. And here 5, 10, 15, 19 with respect to the glucose, that's called actual values. So how we are going to calculate the 19? Because we are going to calculate access point is equal to formula alpha equal to 2 power k by 4. So here to calculate, so here alpha is what here? So 4th root of 2 power k by 4 mean alpha equal to 2 power of k by 4. K is the number of variable. So k is 2 mean what here 1.414, k is 3 mean 1.68. So this is the formula to calculate axial point. So let us see that here how we have to calculate. Let us see 1.6 or 1.8 we consider alpha equal to. Then I can consider how we are going to calculate the 14. So what is the bandwidth what here 5. 5 mean what here I can say. So initial value 15, 15 plus, okay now, and the alpha equal to what here 1.8, 1 unit plus 0.8 units into bandwidth. 1 unit plus 0.8 units into bandwidth what here 5. Mean so 0.8 into 5 what here 4, 15 plus 4 what here 19. Next low value what here 5. 5 minus, you have to subtract it because you have to get extreme low value. 5 minus 0.8 into bandwidth. Bandwidth gap between the lower value and the higher value 5. So 0.8 into 5, 4. 5 minus 4 is 1. So like that same thing is what here? Here 4. Mean what here? 4 plus 1.8 into bandwidth. 0.8 into bandwidth. 0.8 what here? 0.8 into bandwidth 1. 0.8 into 1. 0.8. 0.8 what here? 4 plus 0.8. 4.8. Next what here? Low value are 2 minus. So 2 minus 0.8 into bandwidth. So here 2 minus mean 1 point, okay, 0.8. So we'll get what here 1.2. So like that we are going to calculate. So extreme low value, extreme high value. Please. So I was telling number of experiments to be conducted, how we are going to conduct here. So the chart we have that. So already software we are going to give, but we can understand that. So how you can be able to calculate this total number of experiment by this chart. So you see that up to 14 this year, each experiment has a different set of combination. But only from 16 to 20, we have the same combination. That's called, in order to minimize the error, experimental error, handling error, or any machinery errors. So we are going to conduct a center point 4 times experiments, that is called optimum points here. And this is a quadratic equation. Okay, now. So now my theoretical small, okay, over. Now I would like to start how we are going to analyze using the software, because I have the short video also. Before that, so basically we are going to use the JNTUH software. So this is file, we have that edit. Okay, now. So there we have to open the file, and after that we have the which we are going to use central component. We are going to use it, and we have the box bang and the 3-level factor. A lot of things we have that, but we are going to use central composite. And then, okay, numeric factor, how many numeric factor we are going to optimize? 3 factors are, 5 factors are, up to 10 factors. A category factor we are going to take, okay, by default 0. So here each factor name you have to enter, and the units what you have to enter, low value, high value you have to enter. So like that I was telling. So the numeric factor, 3 factor we are going to optimize, and the name, units, automatically what here? So coded language you will take that what here, what is the low value you have to enter, what is the high value you have to enter. See. Next step, what here? So we have taken, let us see that some studies, they have taken time, temperature, catalyst, the 3 parameters, the okay, important parameter to optimize, and the units is minutes, time, and temperature, okay, degrees centigrades, and the catalyst percentage, low value is 40, okay, high value is 50, and like that they have entered in software. So this is my study. My next slides, what I am going to discuss, how you have to use this software. That is very important. So here, type of, we are going to use the full factorial design types, and the blocks see, let us see that your experiment, you are going to consider you want to design this experiment 2 days, then we are going to fix it 2 days. No, I would like to design the experiment to, okay, now, so split it 3 days, then we can be able to write it 3 days, like that. Okay, we have to enter the 3 values. Next, here, how many response you have? So not only 1 response. So depend upon your study, you can be able to fix 5 up to 5 response also. Here, they have taken 2 response. That is a response. One is conversion. What is units percentage? Activity, what here? So units to activity, you have to enter the IU per ml, what may be, that units you have to enter. Let us see that. So system automatically will give the blocks and lower value and higher value, and 3 factors has given, and the response we have to conduct experiments. Like that, so this run is going to be, okay, now, so we can able to make it fixed run, okay, up to 20 experiments, because I was telling the total number of experiments is what here, 2 power k plus 2k plus n0. So if 3, you take 3 variables, and okay, now center points, you are going to take 6 center points, then you are going to get 20 experiments. So 2 power 3, 8 plus 6, 14 plus 6 center points, that's called optimal level. You are going to conduct experiment, that's called, so 20 experiment you are going to get it. So each experiment, so so take the design chart and you save it, and you conduct experiments. After conducting experiment, before conducting experiment, you have to save it the design chart, and okay, now you have to save it wherever you want to save it, your folder and file. And next is, after that we have to use this previous the design to enter the results of value. So your designs, the coded actual I is telling what here, coded mean -1 +1 0 minus alpha plus alpha is to there. Actual mean what here? Actual, what is the value? Low value, high value, optimum value, that is to be the actual value. So we have to sort out. So in a software, so this is what here coded. See, you see that all coded except alpha value. Okay, now this is all coded chart. So here after that, once again, so when you got results, response 1, response 2, so previous chart you open it and you enter the values, all this in the software. Next, so we have to analyze first how the graphical column, how you get it graph. So with respect to activity, with respect to one variable. So here, so activity versus, so let us see that time. Activity versus once again other parameters. Or so here each response versus time or factors, you can get the different graphs. So here now we are going to analyze the results after entering all the 20 experiments, the response 1, response 2. So first we are going to see how the fit summary. So this we are going to get it. So in that fit summary, so we'll see that left hand is used to that design, status, evaluation. After you enter the result value, you have to evaluate it. After evaluation only, we'll get this tab, that is called analysis tab. That is so we are going to analysis with the 2 response, that is conversion and activity. So conversion point of view, you are going to get this tab, and activity point of view also will get different tab. After finally we are going to get the optimization, that is the numerical optimization or graphical optimization or point prediction, but we are going to use numerical. So remaining slide speaks only with respect to analysis, with respect, okay, analysis in terms of conversion, Okay, now and the activity. So and finally we are going to end up with the numerical optimization. So that is what we are going to see here. So fit summary, next slide, fit summary. Our model is, so our results, it shows the quadratic model is suggested, because so depend upon theoretical value, theoretical model, experiment results will show the, okay, now coherence mean, so it will matches, that's why so quadratic model is suggested. So like that we'll say that here. So in this part here, by this step we are going to get information, our evaluation by analysis you can understand that, so our theoretical model is going to be suggest, okay, match the quadratic model. Next, here we are going to take, so here which mean linear term and 2f1 term and quarter term, here also quarter term is going to be suggested for us. And always we have to get this statement, the lack of fit should be insignificant, that's very important. So lack of fit is negative, okay now, and this is insignificant also negative or positive. So theoretical model and the experimental result is going to fit in maximum, that's why so always you should get a lack of fit should be insignificant. And here same thing is the models, here we are going to get it R square value, adjusted R square value and predicted R square. This difference should be minimal in a quadratic model. So adjusted R square value 0.881 and predicted value 0. So this difference should be always minimal. Then only we can say that so theoretical quadratic model is going to be matched with the experimental results. So here we are going to analysis all the A, B, C parameters, the model terms, and A square, B square, also model terms. So we will take it by before analysis point of view in a software. And finally we are going to get summary, another summary. Here see, always you have to see that each term and each interaction, here each variable A, B, C, A square, B square means cumulative and AB interactive. So which term is important? So with the help of the probability greater than F value, we can see that which value we have the probability greater than F less than 0.05. So that is the important interaction or important term. So here we see that so less than 0.005 mean, so I can say that AC term is very okay, effective interaction term. So to get the best results and optimization process. So here we have to take the what is the R square value. What is the, so R square value in what here? It shows the success rate of this, okay, model to the experimental result is 94%, and the difference between the R square value less than, okay, very minimal difference. So that is the 3 points you have to consider here. One is what here, R square value. So this should be have the maximum. Then what here, the success rate is maximum, okay now, to success this theoretical model to mean experimental result for optimize the process parameter, and this difference should be minimal. So like that we have to take the results, and the final equations we are going to get it in terms of, I was telling y equal to beta 0 plus beta, all constant we are going to here software is calculated by matrices theory. Beta 0 means this value has calculated, 1.03 beta 1 has calculated, beta 2, beta 3 all is with the help of matrix theory it has calculated. Okay now? So these are the chart we are going to see that. And this is a 3D surface chart we are going to see that. Please, you can understand with the help of the video, small video, 2 minutes video. So like that we are going to see the 3-dimensionally how this, okay, each response versus temperature versus catalyst. So two-factor response, two-factor interaction with respect to one response. Two-factor interaction with respect to one response. The two-factor response with the, okay now, third response, second response, like that we are going to get it. And last thing is I was telling the automation, we have to going okay, optimization, numerical optimization. So time we are going to fix in range of, or you want to get target of. Sometimes we have the 3 options here. The range mean, so let us see 24 to 72 hours. So let us see I want to fix to get the 72 hours itself maximum response. Then you fix the target value. The time is 72 hours. Doesn't matter for 24 to 72 hours. Which day is going to get optimized doesn't matter. You put it in a range. So here and like that each variables we are going to have the lower value and higher value to show that, but the two here temperature, catalyst and conversion activity we have to fix depend upon our the target whether we have, sometimes we are going to use target, or sometimes we are going to use a low value, sometimes we are going to high value, sometimes we are going to get what a maximum value. So like that we are going to use. So here the conversion, see the conversion we have fixed what a maximum, we want to get the conversion to maximum. Time should be a minimal. Let us see the time I can consider the minimal, but catalyst I can consider for maximum. So activity also I can the maximum. So like that I can able to fix it. Then I will get, okay, enter, I will get number of solution. So which solution have the higher desirability, that solution combination I will take. Okay now. So what optimized parameter, time is this, is temperature this is, catalyst I will contact the once again experimental results, and this value should be matched for me. So that is the only thing is we are going to have last one video I will show that. With that I will wind up. Please, this is a video I have taken from one. Okay, na? I say a scientist. Please use follow that video. How to use software? Two-digit experiment. The response surface methodology. Hello? Is it audible?
Yes, sir.
We can
It is
hear
audible,
you.
sir.
Okay.
It is audible.
Right. Numerical factors. So they have taken the fruit production, value floor, units we have to enter. It's going to wind up fast, okay? No? First, initially will take time. 2 minutes video only this is. Right, all variables have entered lower units, lower value, higher value, and automatically software will calculate alpha equal to 2 power k by 4, that is extreme low value, extreme high value. And full factorial design. Blocks mean what here, how many days we want to conduct this experiment, 2 days off, 4 days off, 3 days off, they have taken, and followed by center points. Optimum points, 8 times they want to conduct experiments. So 1 day only, block 1. Response they have taken. 5 responses they have taken. Earlier I have shown the slides, okay, 3 responses, 2 responses, but you can take maximum responses, how many responses you want to study. Each response name you have to enter, units you have to enter. So they have taken lightness, firmness, stickiness of their food product. Okay, this is the 5 response they have taken. So like that, design chart they got it. Sort by order, random order or actual order. They have taken the serial order. Then after conducting experiment, they are entering the results. So depend upon how many experiment, alpha 8 times they have taken alpha equal to, so number of variables 4 elements 2 power k by 4 plus. Okay, each variable response they are entering. Right. So all the response they have entered. So this is actual values, summary. Summary means how many response you have taken, how many center points you have taken. Total runs is 32 runs. Because 8 center point they have taken. And 4 variables they have taken. 2 power 4 plus 4 into 4 plus 8, 32. So 4 response they have taken. I've shown that it's a graphical columns, each variable interaction with, I mean, how the response is to be there. Like that. Now we have to evaluate it. So different chart also we can see that. Point view prediction, the evaluation process is going to happen. Quadratic model, polynomial, response, okay, the design response only, the results, see that. We have to get, lack of it should be insignificant here. Graphs, you can see the different graphs here, the perturbation chart, one factor interaction chart, contour chart, 3D surface chart. So depend upon the publication, you can see that. So which publication it has the different, okay, now how they are reported, which graph is important, you can understand, and you can able to take that graph. Next, we are going to see analysis. Lightness, first response. Fit summary. The quadratic model is suggested here. See, lack of fit test should be insignificant. Here also quadratic model. All the terms are model terms only, they kept it. ANOVA summary here should be less than 0.005. So that's significant here. Model is significant. And which term also have the very minimal value, less than 0.005 here, you can say 0.001, 0.001, like that. Lack of it should be not significant. That's very important. See, value, so it's telling less than 0.05, indicate model terms are significant here. So A, B, C square, T square are significant model terms. Values are greater than 0.0, indicate what the model terms are non-significant. Here, show that. Here, R square value has taken, adjusted R square value, predicted R square value, and adequate precision also has taken. So this difference should be minimal. This all calculated. So beta 0, beta 1, beta 1, okay, beta 2, beta, all this thing has calculated by matrices theory. Software has calculated. See, with respect to one response, second response, lightness response, like 3 response, we have the quadratic equation. All we have to copy it to report all data in for publication. So we have to copy it all the constant what we have calculated and paste it in Microsoft. Okay, all the data you have to just copy it. So here, so what are the constant we have calculated with respect to the responses? Here first 3 responses we have taken here, 4 responses, 4 response, beta 0 value, beta 1 value, beta 2 value, like that. You have to copy it for reporting the data. Like that. 4 responses, all constant values, and values are each response, what is the R square value, what is the adjusted R square value, what is the predicted R square value also here for each response. Like that. So each responses, they are going to calculate which are significant terms also. Here, A, B, C are significant terms. So this value you have to take it. This value here, you have to copy it. So last thing is, before last, diagnostic curves. So normal plots versus residual plots. Here, you have the different plots here. Predictor versus actual value. What is your predictor, what is your actual value? Different plots, we have that. With respect to the different responses. Modal graphs, we have that. The modal graph versus interaction lightness response and followed by two-factor interaction. WPC versus barley flour. We have one-factor interaction. Contour plots, 3D surfaces. So here lightness responses. Here WPC is the barley flour. Low value to high value. Cubical plots. Pop-out we also we have that. Like that, each response we have to get the, we are going to get the all fit summary, modals, ANOVA, summary, and diagnostic graphs, and followed by modal graphs. Pop-out we also have like that, each thing, same thing up to 4 response we are going to see: modal, ANOVA, summary, diagnostic graphs, followed by modal graphs. Like that, fit summary, modal, ANOVA, diagnostic cause, and modal graphs. Here, fit summary, modal, ANOVA, diagnostic curve, modal graphs. Like each response, we are going to see that. Lastly, optimization. That is, we are going to see numerical optimization. I was telling in the numerical optimization, you see that we are going to get the goal. See here, they have taken each variable mean. So how many solutions they required? Different combination. We are going to take higher desirability combination. We are going to take WPC. One of the factors in range they have taken. CMC also they have taken range, and lightness response. Now this response where they want to fix it lower value, higher value, target. Let us see here. So they are going to put it a target. So the target value you have to enter it. Right, 42. Next, firmness. Same thing is, the goal is what here for optimization, target. Then they are going to enter the target value. Stickiness. Maximizer, minimizer. So it has to put minimize the value. Simply minimize value. Okay. Next, wellness, maximizer, minimizer, targeter. In range, they are going to keep it. It doesn't matter. Okay. Maximizer, minimizer, targeter. They want to achieve the target. Seven. Then they are going to see the solution now. Solution. Then we will show the different solution here. So each solution, which solution has a higher disability, they should take for the validation. So 0.896 point. This combination has a higher visibility for success rate of theoretical model and experimental results. So value flow has to take this thing. Okay, this is the desirability graph. Copy it. Graph. Like that. Okay. That again, each variable, what is the goal, what is the lower limit, what is the higher limit. All the data should copy it. Okay, the graphs also, the reserved graphs with respect to responses, lightness responses, with respect to the different parameters, A, B, A, C. B, C, C, D. 3D surfaces graphs. Just you have to copy it. Like that, a graphical optimization. Mostly we are going to focus on numerical optimization, but the graphical optimization is there. Point prediction, confirmation, right. Thank you.
Thank you. So since we are in shortage of time, so questions will be posted in the chat box. Please answer to them. Dr. Saida Naik.
Okay.
Thank you very much for the informative presentation on optimization of process parameters by using software tools and models. So it will be very much useful for the students to plan their work. So thank you.
Okay, thank you. Thank you, madam, for giving this opportunity.
Shall we move to the next talk?
Yes, madam. This is the last talk. Mine would be the last one. I'll finish in 6 to 7 minutes.
Okay, ma'am. Okay. Next speaker of this session is our convener of the conference, Dr. Redepalli Uma, who is very active and could able to organize the conference successfully within a short span of time. Congratulations, madam. So coming to our biodata. Dr. Redepalli Uma is presently working as an associate professor at Center for Biotechnology, IST JNTUH. She completed her MTech from IIT Delhi and PhD from Sri Venkateshwara University, AP. She worked as a research associate in IIT and DST Fast Track Fellow at CCMB Hyderabad. She has 26 years of teaching and research experience from various organizations. She was honored with Gates Fellowship in the year 1992 and DST Young Scientist Fellowship in 2005. She received 13 research projects from various government and private research organizations such as Indo-US, BST, DBT, UGC, CSR, AICTE, etc. And she published more than 100 papers in national and international journals of repute, 6 book chapters, and filed 5 patents. Her collaborative projects with various research organizations include University of Florida, HPCL Bangalore, ISCT, NIN, ICRISAT, IIMR, etc. She has guided 12 PhD scholars and 150 MTech and MSc students. Now, I request Dr. A. Uma to present her talk.
Thank you, madam. Yeah, thank you, madam. It's not getting shared. Yeah. I'm going to talk
able to see, ma'am.
Yeah, I'm going to talk on lignin nanoparticles as green bioadhesives. This is basically the work of one of my MTech students. So basically an adhesive is a substance which is used to join 2 or more parts so as to form a single unit, something like a Fevicol that we use. Different types of adhesives, natural adhesives, thermoplastic adhesives, use thermosetting adhesives and rubber adhesives. So this is based on the type of application. We can classify them into different types of adhesives and the material that is being used for that particular application. Though we feel that adhesives are a small entity in the market in biotechnology, but they have a huge market. As far as the commercial aspects are concerned, they have a huge market. So if you just look at the Indian adhesive market size, it is going to grow up like anything. So there is every need to, we cannot underestimate the commercial value of adhesives. So as many possibilities as possible, we have to explore to make the adhesives. So basically we are using phenol formaldehyde adhesives so far for the plywood applications. And this is basically derived from the petroleum based products. And the main advantage of this is the low initial viscosity and strong water resistance, which is very much required, excellent thermal stability. Not only for the normal wood adhesives, we can use it for shippings and all these things also. That also requires wood adhesives. So the advantages are, this is basically a petroleum based product, expensive, and because it is coming from petroleum based, it is non-renewable, and it releases free aldehydes, which is a potential 2A level carcinogen. So it can cause some environmental issues. So there is every need to look for some of the bioadhesives. So what are the bioadhesives? Bioadhesives are non-chemical, they are chemicals, but they are derived from the biological sources. And we have got saccharide based, protein based, aromatic based, and oil based, and other types of adhesives. Next. So for the key drivers for the bioadhesives, why we have to opt for bioadhesives is the recent governmental legislative changes to minimize the health effects relating to product emissions of volatile organic chemicals, the use of renewable materials as cost-effective replacement for any type of petrochemical-based adhesives, increased concern, of course, towards the human health, and of course, the depleting sources of petroleum resources. And price volatility of, that is the constant problem that we face with the petroleum-based products, and adhesives is one such thing. And the challenges and the opportunities that we have is bio-based adhesives with the same bonding performance and same cost are lower than synthetic adhesives. Sustainable adhesives should not only be available at low cost, but also need to be easily distributable, fast reacting, and have long life. Key factor is to achieve the same production speed. So if you just look at a plant cell wall consists of cellulose, hemicellulose, and lignin. In fact, lignin in every plant cell, if you take, this lignin is the adhesive material that combines the cellulose and hemicellulose strands. So without lignin, there won't be any mechanical strength to the plant, and this also gives some protection from the microbial attacks. So if you just look at the structure of lignin, it is basically something like a phenol only. It has the phenol structure. Phenol has C6H5OH, but this also has a C6 ring, but some substituents are there. So this is because of this structure, it can be a substitute for phenolic based resins, and it is non-toxic and it is available plenty. So it has same properties as that of phenolic compound. So we have got different types of lignin based on the type of source from which we take it. So we have guanacil, syringyl lignin, and hydroxypropane type of based lignins. So the advantage of using lignin is, only thing is it can be a very good substitute for phenol. But if you just look at the aspects of bioadhesives, an adhesive should offer very high surface area, and it should have very good reactive groups. So these two groups can be improvised from the lignin by making that into nanostate. So a higher specific surface area is achievable when we bring that lignin into the nano size, and it can have better porous structure, and better chemical reactivity is available when we bring that into the nano size. So this in fact improves the adhesive properties of that. So we can just take the lignin from different types of what my student has taken is from the black liquor, and he has divided that into lignin and the remaining solids, precipitated the lignin, and that lignin he took for the preparation of nanolignin. So these are some of the images of alkali lignin and the nanolignins. So where we can find the surface area increase. The specific surface area of lignin has improved by about 10 times by bringing down the size of the lignin. The pore volume has also improved. The specific surface area, improved specific surface area is responsible for the betterment of chemical reactivity with formaldehyde. So we have estimated the particle size distribution of nanolignin using DLS scattering method, and FTIR is used for the characterization of the nanolignin. And this nanolignin for the preparation of the phenol formaldehyde resins, we have taken we have used a little bit of molten phenol also. This nanolignin phenol adduct is combined with formalin in the presence of methanol as a catalyst at sodium hydroxide and reflexed it, and then we prepared the adhesives. So next thing is what we did is we have tested the strength of nanolignin phenol formaldehyde resins, because once we try to combine, that should give very good strength between the two plants that we add. So this type of SCI is very much required, because when we are using in some ships and all, that ships and all will be carrying heavy loads of materials. So if the adhesive that is used is not good enough, that can lead to some of the problems related to strength. So we have to look at the strength of this one, and this nanolignin one has improved, has very good strength, and it is about from 2.4 to 2.9 megapascals. So this is how we try to look at the strength. We try to pull these blocks of wood that has been for which this adhesive has been used, and it should not be in a position to lose out easily. And that tensometer missions will be used, and we will try to see at what pascals, at what megapascals they are able to resist the looseness. And this is the FTIR spectra of different types of phenol formaldehyde, nanolignin and normal lignin. And we can see that nanolignin has better comparability with lignin, phenol formaldehyde. So again, we did the TGA curve. So TGA curve is used to see, like when we are trying to melt the material, we have to see how the material properties are changing. So what is the temperature stability that the compound can offer. So we have to do the TGA curve, and TGA curve, the nanolignin based one is a better one and comparable with the phenol formaldehyde resins. So in conclusion, in view of the increasing concern towards human health and shortage of petroleum resources, the development of green and low toxicity bioadhesives has attracted much interest and economically viable. The future will be an environmental friendly bioadhesive based natural and renewable resources and produced under sustainable conditions. Thank you.
Thank you, ma'am. Thank you very much for a very good presentation on lignin bio nanoparticles as green adhesive, which is environment friendly. And thank you very much for completing the talk within a short time.
Yes, yeah, madam.
So with this, shall I conclude, ma'am?
Yeah, please.
Trisha? I conclude the session by thanking speakers of this session, Dr. Kalpana Shastri, Dr. A. Uma, and Dr. Saida Naik. And I congratulate all the speakers for their excellent presentations. I'm thankful truly for the inspirational, research-oriented, and informative talks useful for the biotechnology and bioengineering students. I thank Dr. Uma and her team for giving me this opportunity. Thank you.
Madam, in fact, we are thankful to you for gracing as a chair. Thank you so much, madam. You have spent so much of your time with so much of patience. Thank you, madam.
Thank you. Bye.
We will, Lalita, we will join at around 3 o'clock. We will go for lunch. Lalita?
Okay, madam, we can wind up for now, and I can start around 10 to 3.
Yeah. Okay.
It's
Thank
already 2:40, madam, now.
Yeah, it is already 2:40. We will join at 3 o'clock. Is that fine?
Okay, ma'am.
Okay, ma'am.
Okay. Okay.
అరే ఇప్పుడు నెక్స్ట్ సెక్షన్ 4 కూడా ఏమన్నారు మేడం? ఏం కాదు.